We on? Check, check. I think we're good to start. Hello. That's the enthusiasm I'm looking for. Then I think we are going to kick it off. Hello, everybody. My name is Storm Greenway. I'm chairman of the PMD Foundation. Still have sunglasses around my neck. Thank you all for joining us today. We're really excited to just get together and have a gathering, all of us here in the community and everybody who's online as well. Thanks for joining us. We have a whole lineup of great speakers, and then myself, great speakers, and myself. I want to make the distinction. But I appreciate you all being here, and yeah. So let's talk through the agenda right quick. I know this has been posted online a few times, but we are going to be led off by Julia Porcari from CHOP. We are then moving to a dear friend of mine, Grace Hobson, talk about some genetics. I'm going to be talking through the family planning from a non-scientific side with our friend Doug Harder, who will be remote in. After that, my dear friend Courtney Bisbee from Ionis is going to be talking us through the rocket and orbit trial and study, would be the right word to call it. After that, going to be talking a little bit, actually we can omit that 3:15 to 3:30 because Grace is going to be covering that. Ashley Dyke from the LCC Foundation and myself will be talking through some patient advocacy. And after that, we will have a great panel of caregivers all here together to talk a little bit about the experience, led by one Melanie. Round of applause for the undertaking. And with that, I think we're good to kick off. Julia, hand it to you. Actually, I won't because there's another microphone. Wonderful. And green button to move forward.
Perfect. Well, hi, everyone. I am really excited to be here. For those in the audience that I don't know, my name is Julia Porcari. I'm a child neurologist at the Children's Hospital of Philadelphia and a research fellow. And I was asked to come and talk a little bit about what we think the current research landscape looks like in both PMD and PMLD. As you might imagine, that is an ambitious undertaking. So there will be a couple of points where I am a little bit more cursory, so go less in depth, but my hope is that I will set the stage for a couple of the other speakers that are going to talk, and also happy to take questions. And if you can't hear me at any point, let me know. All right, I don't have many disclosures. I'm supported by a neuroepidemiology training grant at CHOP, and also I'm a neurologist on Ionis trials for Alexander disease, and then upcoming PMD shortly, since we were just started up as a site recently. And this is a brief agenda. So again, I'm going to be covering two diseases, both PMD, so Pelizaeus-Merzbacher, that you're all very familiar with. I'll do a quick review just to make sure that we're all on the same page, describing what we recognize as a long recognized PMD, hypomyelinating disorder. And then I'll briefly talk about research. And then I'll do the same for PMLD, which is a disorder that I study a little bit in more detail. And then I'll wrap up with a quick review of some current clinical management best practices, which I think are applicable across both of the disorders. Again, PMD is a disorder that most of you in the audience are intimately familiar with, and you probably also know that it's one of the leukodystrophies that was one of the more common leukodystrophies, and also one that we've known about for the longest. So it derives its name from one German physician, Friedrich Pelizaeus, who first described a family back in the 1800s, really. So a family of boys who had nystagmus, developmental delays, and spasticity. And the second part of its name actually comes from another German physician, Ludwig Merzbacher, who was the first person to describe what these boys unfortunately had in terms of pathology in their brains. Also, the first person who gave us a sense of how this disorder was passed on from one generation to the next. The gene was only described about 100 years later, but still 1985, so quite some time ago within the context of genetic disorders. So they were able to identify that PMD seemed to be caused by variants or mutations in this one gene, PLP1, which is located on the X chromosome. And since then, we've learned quite a bit. And so I think of PMD as what I'll call a canonical hypomyelinating disorder. So I'm sure that at this point you've learned that leukodystrophies come in many different flavors, and two big buckets that we think of are demyelinating disorders and hypomyelinating disorders. So demyelinating disorders, such as MLD, for instance, or ALD, are disorders in which myelin is formed and then destroyed over time. Hypomyelinating disorders are disorders in which, for multiple reasons, myelin just does not form normally. And so what is myelin? So myelination is a normal process by which neuronal axons, that blue cell that you see in the middle, are ensheathed, so they're covered in this fatty coating. And that coating provides insulation. And insulation is critical in the central nervous system because it makes communication between cells faster, and it actually also supports the health of the neuron itself. It's a process that begins in utero, so very early on, and we think is largely complete by about 2 years of age. This is another cartoon that is showing you a little bit more detail. So that purple cell that you see there, that's an oligodendrocyte. So these are the cells in the central nervous system that are the myelinating cells. And what you'll see, and do I have a pointer? Yes, perfect. They have these processes, so these almost appendages that wrap along the blue axon. And this is actually a cross-section of what you might see on an electron microscope. So that light blue center that's been colored in, that's the axon. And then these almost onion rings, those are layers and layers of myelin that are wrapping along it. And we know that PLP1, the gene, encodes two proteins, PLP, proteolipid protein, and then DM20. And we know that they represent a large proportion of the protein that's in myelin, about 50%. So definitely doing a lot of heavy work here. And we don't know exactly what all those roles are. This is an area still of a lot of bench and preclinical investigation, but we know that they're very important for what is called a compact myelin sheath stabilization. So they make sure that all those onion rings stay nice and tightly wound around the axon. Since the first discovery of the gene in 1985, we've learned a lot about what types of genetic variants cause PMD and what the mechanisms that are responsible for going from genetic change to then problems in the cell to then the phenotype or the symptoms that our patients have. We've learned a lot. There's still a lot that we're still learning, but a large proportion of patients that have variants in PLP1 have so-called duplications. So they have too many copies of PLP1, and too much is not always a good thing in biology. And what we know in these patients is that, let me see if I can go back. What we know in these patients is that when PLP is transcribed from DNA in the nucleus, it goes into the structure called the endoplasmic reticulum, where it's converted from RNA into protein, and then it's supposed to go into these vesicles that then go all the way out to the membrane that's myelinating. We know that when there's too much PLP, that PLP gets stuck in these vesicles, and it also traps other proteins, other lipids. And so you can imagine all of these beautiful myelin ingredients are just not able to migrate all the way out to myelin sheaths. And so that's clearly one mechanism of disease. You can imagine also that when you're gumming up the cell with proteins and lipids in a spot that shouldn't be there, there can be other downstream effects. And so we know that there's oxidative stress. So oxygen is incorporated and changes chemicals in the cell. And that also is associated with inflammation. So microglia are the inflammatory cells in the brain and seem to be dysregulated in a number of patients. Another big category of genetic mutations that we'll see in PMD are instead missense variants. And so these are patients who don't necessarily have too much PLP, but have abnormally formed PLP. And so what happens in this case is, the RNA is transcribed still in the nucleus, goes into that endoplasmic reticulum again, but the protein is misfolded. And there is actually a phenomenon that's seen in many different disorders that have this process, so not just neurological disorders, not just leukodystrophies. That is stressful, we think, to the endoplasmic reticulum. And there are multiple downstream effects there too. So again, we have formation of those reactive oxygen species. And then we have a system called the unfolded protein response, which is this big complex system that the cell has to recognize proteins that are misfolded and try to respond to it. And it does that in a lot of different ways. There are proteins called chaperones that try to refold the proteins. There are proteins that target those misfolded proteins to degrade them and get them out of the cell. And then there are other strategies like trying to slow down proteins so you don't get these misfolded proteins sort of piling up and gumming up the system. And if that doesn't work, unfortunately, the cell actually decides that it's time, and so undergoes a process called apoptosis, and so it dies. So a lot of potential mechanisms. And it may be because of all of these different variants. And, I introduced 2 categories, but we have frame shift and splice size variants and null variants, many different types of variants in PLP1 related disorders. And again, it's probably because of that variability that we see such a huge spectrum of disease when it comes to variants in PLP1. And so we've talked about PMD a few times. Many of you likely know already that even PMD in and of itself comes in a lot of different flavors, right? There's conatal PMD, there's classic PMD, transitional in between. And so these are children typically that are presenting early on with eye movement abnormalities called nystagmus, hypotonia, so low motor tone, and then often spasticity as they age. But on the other end of the spectrum, we have young adults who are presenting with spasticity mostly, and then difficulties walking, oftentimes autonomic dysfunction and bowel and bladder problems. And along the way, there is variable involvement of cognition and speech, but it's a very broad spectrum. It's also very messy, I think, as we have been trying to understand this disorder more. So with that all in mind, just to make sure we're on the same page, let's move into talking about research. So this is a heat map that was put together relatively recently out of a conference that was held. So I don't know how many people in the room are familiar with GLIA. So it stands for the Global Leukodystrophy Initiative. And it's a consortium funded by the NIH of both researchers and advocates across the country with international collaborators as well. And the mission of the consortium is to advance what we call clinical trial readiness. So how do we get diseases studied, well understood, and on the pathway to having disease-modifying interventions? And this network has recurring scientific conferences, and the last one was actually this past February. And the goal of this conference was to try to get a snapshot of where the research landscape or where research was for each of 23 diseases, actually. And so what we did for this particular conference is before the conference met, we had work groups that were put together for each one of these diseases. We gathered ahead in meetings and had every participant complete a survey to give us a sense of what they thought as an expert in their particular leukodystrophy, what they thought the relative strengths and then gaps and weaknesses were. And then we had all that data brought back to the conference to discuss it together. And so this is a heat map where orange is highlighting areas of gaps, blue or teal are relative strengths. We had the survey structured in 2 big domains. One was, what do we understand and about the clinical phenotype and how are we managing patients sort of in the now? And then the other domain was, what are we doing to get new disease-modifying therapies to patients and where are we along that trajectory? And each one of these columns is a different disease. And right there in the middle is PMD. And this was a pretty large work group. It was a combined PMD and PMLD work group with about 20 participants. And what you might notice is that there's a lot of teal, so a lot of relative strengths. And so advocacy came out as something that researchers thought was a strength. Clinical trial planning and ongoing preclinical work, as well as symptomatic management. You'll also notice, though, that access to disease-modifying therapies obviously is more in the orange. There's a lot more that we need to do there. And so these are some of the topics that emerged in the actual sort of concrete discussion, and I'm going to talk about a couple of these, but not all. So I'm going to focus mostly on natural history and then a couple of disease-modifying therapies. Some of this work was done by the GLIA group. The latter point is from the research literature more generally. So if you do know about GLIA, you also probably know that we run a really large natural history study. We call it a real world data natural history study. And what many of you have likely contributed to is collecting medical records, which we, with an army really of a big team of research coordinators, comb through to try to get a sense of what the onset of disease looks like and then how it evolves over time. We have a large cohort, and this is a smaller subset of that cohort of 105 patients. And what this data allows us to do is to get a sense, for instance, of how patients present. And so as you might imagine, for our PMD cohort, the vast majority of patients are, when they are first having their first manifestations of disease, come in with abnormal eye movements, with developmental delays. Those are seen in over 75% of children. But there also are other very common complication or early signs, so delays in cognition or speech, as well as stridor, which is a symptom, for instance, that's well recognized in connatal PMD, but all the way down to even autism spectrum disorders and seizures really at first presentation. So a wide variety of presenting symptoms that can be present in different combinations. And another thing that we look at in this data is what was the timeline of milestone acquisition. And this is really critically important when we're thinking about designing designing clinical trials. We need to have a really good understanding of how development is proceeding in children who may have disease. Because, and Dr. Vanderver, if you've heard her use this analogy, talks frequently about the fact that leukodystrophies are trying to modify the trajectory of development, but we need to know exactly where that trajectory is to be able to then see if a potential intervention has modified the trajectory away from what the disease itself would be doing. And so what you see on this particular graph is a list of 18 different milestones on this side. So these are milestones for which we have normative data, which means from very large studies, we know what the typical age at which children acquire these milestones are. And they're grouped in fine motor, so movement of your hands. They're gross motor in the middle, so sitting up, walking, rolling, things like that. And then at the bottom are social and language milestones. And at the bottom, on the x-axis, we have age, and every dot represents a patient. And then the blue bars that you might see, they're a little faint. That is the age range at which typically developing children will acquire that particular milestone. And then the two shades of orange and red are areas that are delayed, again, based off of what we know in typically developing kids. And so what you'll see is that especially for gross motor milestones, many of those milestones are shifted over. So children with PMD are typically acquiring these milestones late if they do acquire them. And that's less evident actually in the language milestones that seem to be more on time, and perhaps that resonates with some of your experience as well. Other things that we can do is look at the number of children who acquire, don't acquire, or lose milestones over time. And so these graphs are, these are head control or sitting without support, walking with assistance, walking independently, 1- and 2-word phrases. And the dark gray is showing you, so the x-axis is age in months, the y-axis is count, and the dark gray sections, these are children who acquire a milestone at that particular age. The gray are children who have not acquired a milestone at that age, and then the blue are children who've acquired and then lost a milestone. And so this starts to give us a sense of, again, what do we expect to see in the natural history of PMD without us, beginning any interventions. And I mentioned, again, understanding the natural history of disease is really, really important. And one thing in particular, in addition to just knowing what we're comparing ourselves to with an intervention, another thing that we look for is stratification. And so that has two purposes. On the one hand, it's really important, as you know, to families to have a sense from their doctors of what can I expect for my individual child. Again, PMD has this wide spectrum. Where is my child? What can you tell me? So that, of course, is crucial. And then also when you're thinking about clinical trial planning, I want to make sure that I'm not comparing apples to oranges, right? I don't want to put together a patient who may have more connatal PMD with one who may have more spastic paraplegia, because their courses are going to be very different. So I need to be able to do that well. And what we've learned as the spectrum of PLP1 disorders has widened is that those historic, conatal, classic, transitional categories are helpful perhaps on a population scale, but not terribly helpful actually when you look at the individual patient. So we need something better. We've also actually tried to do that same stratification based off of classes of genotype. So do you have overexpression or null or loss of function variants? And based off of what we know now, and that I think is an area that will continue to grow, and I'll mention it a little bit later, but that also type of classification doesn't seem to work terribly well. And so what we've done as a group recently is try to get a sense of whether early developmental milestone can help us identify those subgroups. And so specifically, we looked at stridor as a presenting symptom. So when a child first has manifestations of PMD, do they have stridor, which is this particular noisy breathing that some children with hypotonia of the larynx and pharynx have? And then also head control. And so based off of that, we can estimate probabilities of gaining skills like being able to sit independently, being able to use a thumb and finger grasp, or being able to speak one word. And when we bucket our children in 3 buckets, so buckets of children who had stridor when they first presented, buckets of children who did not have stridor and acquired head control by 12 months, and then an intermediate bucket of children who did not have stridor but also did not acquire head control by 12 months. We see, again, this is plotting those same developmental milestones with counts on this side. And dark blue is number of patients that acquired that skill on time. Light blue is the number of patients who acquire that skill, but in a delayed fashion. And then dark gray are patients who never acquire those skills. We see that these 3 groups are very different, right? There's a lot more blue in this group of children who don't have stridor and acquire head control by 12 months compared to those that had stridor, for instance. And so we think this might be a better way, perhaps, of bucketing children to both give, I think, some prognostic information to families, as well as think about stratification in the context of trials. This same classification can also be helpful when you're thinking about medical complications. So these are called survival curves. So on the x-axis, you have the age in years. On the y-axis, you have cumulative events. So these are for G-tube placement and time to scoliosis. And so every time a child at a particular age has an event, that curve goes up a tick. And I'm showing, again, 3 groups. So the green is, again, that no stridor head control by 12 months group, and the red is stridor presentation, with the orange being that intermediate group. And it's a little bit hard to see because it's small on the slide, but you trust me, these curves are separating, which is what we try to see. Does the time and the frequency at which these complications happen differ between these 3 subgroups? And the answer is yes. All right, so that is a quick update on some of the natural history study that we have going on. It's a manuscript we're currently working on publishing, so hopefully that'll be available relatively soon. And then I just briefly wanted to mention a very, very large project that we have going on, trying to do a better job at genotype-phenotype correlation. So this is a project that has been in large part spearheaded by Dr. Hobson and combines her data set from Nemours with our GLIA data set, as well as patients in the literature. So over 300 mutations, if I'm not mistaken. And what we're trying to do here is develop a better system for classifying variants and doing some of that prediction that I've told you so far is hard to do. So this is also a manuscript that is in preparation and hopefully will be out relatively soon. All right. I won't talk about the Ionis trial. I think Ashley will to some extent, so I'll leave that to her. But I will briefly talk about 2 other disease-modifying interventions that are currently being evaluated and are in different stages of that clinical trial process. So the first is deferiprone. And the basis for this is a knowledge that in PMD and PLP1 related disorders, there are disturbances in oxidative stress and reactive oxygen species, as I mentioned earlier, and also a knowledge that iron metabolism plays a role in reactive oxygen species. So having Iron is normally present in the body. It's really important to allow you to carry oxygen to your tissues, and its metabolism is very tightly regulated. And when things go awry, you can have complications. So oxygen species are added to chemicals and molecules that they shouldn't be added to. And one big category of molecules that we worry about are lipids, right? So protolipid protein, the lipid in myelin, they're a vulnerable species. And there's actually a form of cell death called ferroptosis that's caused by abnormal iron metabolism. And we've known at this point for some time that iron metabolism is dysregulated in PMD. And there was a group several years ago that looked at using iron chelators. So these are compounds that trap iron. And they have looked both at oligodendrocytes that have PMD-related variants, as well as mice that are mice models of PMD, and were able to show that when they used there was 2 different chelators, but differentiaprone in particular, that amount of myelin that was produced in these mice was higher, and there was less inflammation, so less of that microglia activation that I'd mentioned earlier. And so this has actually been the basis for a phase 2 trial that was recently announced and is set to open in the Netherlands, that at this point is restricted to patients who live in the Netherlands and are able to frequently come for visits, and also children who have classic or conatal PMD. But I'm really excited to see if this is something that might be useful and helpful for patients, and hopefully if it is, that would be something that would be expanded. And then the second disease-modifying therapy I wanted to briefly touch on is the ketogenic diet. So we've talked a lot about lipids, and you've probably heard about ketogenic diet as a high-fat, low-carb diet that is actually approved and known to be helpful in children or adults even who have epilepsy. The mechanism is very different, but the thought was, if we change the lipid input in the diet, could that also have some potential benefit? And there were some, again, mouse model based studies that were done and published several years ago at this point that showed, again, this is a mouse model, the GIMP model, that ketogenic diet seemed to restore the number of oligodendrocytes. So again, oligodendrocytes are those myelinating cells in the central nervous system, improve myelination, and actually improve motor function. And so these are some graphs that are pulled from that paper. What you see here is bars that are quantifying the number of oligodendrocytes in the cell. So this is a normally developing healthy mouse, and these are transgenic mice that were fed a regular diet or a ketogenic diet. So the bright green are the animals that were fed the ketogenic diet, and you'll see the numbers of oligodendrocytes there are pretty similar to the white bars of the healthy animals. And these are 2 graphs that are representing motor function. So this is an animal on a rotating rod. Are they able to stay on that rod, or do they fall off at different weeks of age? And this is animals are put on a pole, and they have to try to come down the pole in a controlled manner, and they quantify how many times the mouse slips. So those are 2 assays that are very commonly used in bench and mouse-based research. And so based off of that improvement, the group in Milan is doing a pilot study with just a few patients to see if the ketogenic diet works. I think at this point, I talked to them earlier this week. Sorry, I keep on moving in and out of the microphone. I apologize. They've treated 3 patients, and we'll see, again, what that data shows. I will say there's a lot of interest generally in even popular culture on the ketogenic diet. It's a really hard diet to implement, even for my patients who have epilepsy, and it comes with potential side effects. So I think exciting if there is potentially some benefit, but it's not something that I would recommend for every patient at this point. So just a word of caution. All right. I know I'm over time already. And I apologize, but there's, as you can see, a lot to go over, and even more, honestly, that I could talk about. So PMLD, the second disorder in our odd sort of collection of PMD, PMLD disorders. So this is a name that was first introduced when clinicians started to see patients that very much looked like they had PMD, based both on their clinical presentation and their MRI scans, but had some features that clearly set them apart. So these were patients that had that phenotype but did not have PLP1 variants, so clearly did not have a PLP1 related disorder. And also had autosomal recessive inheritance. So it was a disorder that was seen in girls as well as boys, which is, again, something that we don't see in PMD, with the exception, obviously, of carriers, which is a whole other big conversation to have. And then the other thing that was different is that if you look at MRIs of patients with PMLD, there's a lot more involvement of the brainstem. It's a little bit hard to see, I think, in this figure, but the brainstem and the pons in particular is much brighter. We identified the gene that was responsible for a large proportion of these cases, relatively speaking. Everything in genetics is rare in small proportions, back in 2004. And we know at this point that within that original, somewhat heterogeneous, so vague cohort of PMLD, a relatively, again, large percentage are accounted for by patients who have variants in these genes. And a quick note on terminology in case you're confused. In medicine, and leukodystrophy medicine in particular, we change names often. And so you will see folks in the literature refer to PMLD as PMLD or PMLD1 or hypomyelinating leukodystrophy 2, or my personal favorite, GJC2 related hypomyelinating leukodystrophy. It just makes it clearer what gene you're talking about. You'll also, if you're curious, notice that the name of the gene has changed. We call it GJC2 now, but for a period we called it GJA12. So it's confusing, but GJC2 I think is what we're sticking with for right now. All right. So PMD is a disorder of myelin structure and myelin component structure. PMLD is a disorder of gap junction biology. So GJC2 encodes for a protein called a connexin. And connexins are this very large family of membrane proteins in glial cells. They're each 6 connexins actually come together on the membrane of one glial cell, so typically in oligodendrocytes, but astrocytes as well, to form what's called a hemichannel. And then one hemichannel on 2 different cells form what's called a gap junction. And these gap junctions allow for what's called electrical or metabolic coupling, so really communication between cells that otherwise aren't able to communicate with directly. So they can pass electrical signals, they can pass nutrients across to each other. And there are many neurologic disorders that actually have this system. So Charcot-Marie tooth, if you've ever heard of that, and then another leukodystrophy, oculodigital dental dysplasia, is also a gap junction biology disorder. There are many, many different oligodendrocytes. And again, the one that seems to be the problem in PMLD is this connexin 47. I should mention there are different combinations of connexins. Connexin 47 is oligodendrocytes and then combines with another one, with another hemichannel formed by CX47 on oligodendrocytes, and then has another partner on astrocytes, but different combinations for different cells, which is just generally how biology works. Alrighty. Okay. And we know that in PMLD, these proteins either don't make it to the surface of the membrane, or they make it, but they don't form normally functioning pores. So they don't allow for that normal electrical or metabolic coupling between cells. Okay. Since it was described, we've also learned, right, it's a leukodystrophy, and leukodystrophies present along a spectrum, that PMLD also has a similar spectrum. So similar to PMD, there are children who present typically before the age of 6 months with nystagmus, hypotonia, spasticity over time, developmental delays. And then at the very other end of the spectrum, there are patients who instead typically present in adolescence or young adulthood with a spastic paraplegia presentation. So also gait dysfunction, autonomic dysfunction, bowel, bladder problems. And then more recently, there have actually been a couple of reports of patients who have no clear neurological symptoms, but had MRI scans for other reasons, and who have evidence of hypomyelination on their scans. So I think we're still in a phase where we're learning a lot about the spectrum of this disorder. It's a younger disorder compared to PMD. Okay, so same exercise as PMD. And you'll notice that there's more orange here, right? So still, I think relative strengths and advocacy, I think with the joining of PMD and PMLD with the PMD Foundation, that's obviously been, I think, a big development. But a lot more to do, I think, in terms of understanding how this disease works and ultimately developing clinical trials. In terms of specific research updates, I'm going to present to you a little of my work actually on natural history of PMLD, and then briefly a couple of slides on some gene therapy approaches that are currently in the works. Okay. So we have a much, much smaller cohort of PMLD. PMLD is much less common than PMD. So I have data from about 25 patients, and here boys and girls, again, because it's a disorder that affects girls as well. And what you'll notice, this is the same presenting features graph that I showed earlier, is that these patients tend to present really mostly with nystagmus. So we don't see as much of the tone abnormalities, at least early on. That changes quite quickly over the course of toddler years typically, but not as much stridor, really no stridor in our particular cohort, no seizures, no early cognitive delays, so a little bit different of an initial presentation. Despite it being it's not exactly the same. And this complicated graph is a version of something that I showed you earlier. But again, what I'm showing you are those 18 developmental milestones. Dark blue, again, is number of patients that acquire that particular skill on time. Light blue in a delayed fashion. The dark gray are patients who never acquire that particular skill. And the lighter gray still are patients for whom we just don't have that data. Medical records, it turns out, aren't designed to do research, even though we use them for that purpose. And then what you'll see in panel B is, again, the age at which children are acquiring that particular skill. And also for orientation, again, it's fine motor at the top, gross motor in the middle, and then social language at the very bottom. And if you still remember the PMD slide from earlier, you might notice that there's a lot more blue, generally speaking, in this gross motor bucket. So children with PMLD tend to acquire early milestones up to actually walking with assistance much more frequently than patients with PMD. Similarly to PMD, there's relative sparing, so there's less delays when it comes to language and cognitive skills. So that's more of a similarity. And then again, we can look at the number of times or the number of patients who lose that skill and when they lose that particular skill. And loss in this graph is magenta, the dark gray is never loss, and then the lighter gray is either not applicable or we just don't have the data. And you'll see that a large proportion of the patients that had acquired a particular skill loses that skill, especially with in the gross motor domain. So 50% of the children who learn to walk independently will lose that skill. And if you look at a median age, this is across all domains, that first skill loss tends to happen at around 6 years of age, at least in our cohort. All right. So this is the clinical data. Just briefly wanted to present some of the preclinical data, again, not mine, but from the literature, just so you have a sense of what's going on. So earlier, actually, during the conference, we had a researcher come and talk about a lot of beautiful work that's being done on understanding how variants affect the protein structure. So I think there's a lot that we're going to learn from that. I don't have any slides on that, but I think there's, we're understanding how the disorder works a lot better. There's a lot of progress being made there. There's also a group that's been working on gene therapy. So this is an adeno-associated virus vector that at this point has just been studied in mice that have GJC2 variants as well as variants in another gene. It's hard actually to have a mouse that looks like PMLD. There's a lot of work being done in refining that mouse model. But what this slide is showing you is animals that were treated with a placebo vector, so placebo gene therapy that didn't have the gene in it, just had the additional extra components. And then these two slices, this is immunohistochemistry, are instead showing you the fully functioning gene therapy. And dark black is what myelin looks like, and you want to see myelin that's arranged almost linearly. So and this is a part of the brain called the corpus callosum, which is a big bundle of white matter in both human and mice brains. And so what I hope you can appreciate is that there's a lot more black on this side of the image, a lot more white. These are they're called vacuoles. They're areas where the myelin is sort of falling apart. So very different there. And they've quantified this in a number of different areas. So the corpus callosum, parts of the spinal cord, the internal capsule. I should mention this was a gene therapy that was delivered directly into the brain of these mice. And then they also looked at motor function. So these are actually mice footprints, and these are animals that were treated again with a placebo version of the gene therapy, this with the actually functioning gene therapy. And what they can use this to do is to look at stride length, so how long are the steps of these mice, and they also can look at how far apart are their paws when they're walking, which is a measure of ataxia, so imbalance. And then these are some of those same motor tests that I showed earlier. So how many times are they falling off the rotor rod? Are they slipping when they're trying to walk on a beam? And these are all animals with disease, but animals that are not treated or treated with placebo compared to the ones that are treated. And these are significantly different, which is encouraging. So hopefully we'll see how that progresses. And so there's a lot of work being done, ranging really from bench all the way to early clinical trials, definitely in PMD. PMLD is not quite there yet, but hopefully will be soon. So we certainly don't have a cure. And I think that's a really hard place to be in, both as families and also as physicians. And I think oftentimes, especially in that first meeting, there's this sense of, okay, we don't have a cure, so there's nothing to do, right? And my resounding answer is no. There's a lot that we can do in this sort of in-between space. And so I just wanted to remind us all of that. And this is, I think, something that you're all as families keenly aware of. But I like to think about symptomatic interventions in leukodystrophies generally, but especially in leukodystrophies like PMD and PMLD. I like to think of symptomatic management as being standing on three pillars of sorts. So what we're working towards is maximizing developmental potential. So how do we get the most out of the myelin that's already there, or promote the amount of myelination that is possible for that particular patient as much as we can? I also like to talk about maximizing function. So how can we potentially adapt the environment to, again, maximize quality of life? And then also minimizing medical complications. And so when it comes to maximizing developmental potential, I think about therapies, enrichment, nutritional support. We know that nutrition is really, really important for healthy brain development. And then for maximizing function, I think also about therapy, but adaptive equipment, and then school support. So how do we keep children in the most integrated form of schooling that works and benefits them, so that they can get that social experience and enrichment too? And then the minimizing complications is often where the doctors come in, and certainly the neurologist perhaps a little bit more. And so my job as a neurologist is to help treat spasticity, dystonia, seizures, which are relevant for many of our children. And then also we partner with other specialists to think about preventing things like fractures or scoliosis, aspiration, urinary retention, and UTIs. And I mentioned partner because I think what's absolutely critical in leukodystrophy care, and in PMD and PMLD in particular, is building a multidisciplinary care team. So it's not just the neurologist or not just the geneticist, it's the neurologist who hopefully is talking to the orthopedic surgeon and the PM&R, the rehab doc, and a urologist if they're on board too, to make sure that your plan is integrated and really meeting the goals of that particular family and patient. And so with that, again, I'm sorry that I went over, but hopefully that was somewhat helpful. I'm happy to take any questions if we have time, but Storm, you can comment.
If anybody has any questions, feel free.
Great.
Give me the mic.
I think there's a mic coming your way.
Just that way the people who were watching this on the stream can hear it.
Yeah, so I have a question.
Yeah.
There's PMD and PMLD.
Yeah.
Are they mutually exclusive genetically?
They don't have to be, theoretically, but I don't know. We haven't seen any patients who have both, as far as I'm aware. Okay.
My youngest son has a trach. And
Okay.
the reason why he has a trach is tracheomalacia.
Mm-hmm.
And when we discovered that he actually needed the trach, and not breathing 100 times overnight and all that because of obstructive apnea. So first the doctor was like, we can break his jaw and reset it, we can clip the tongue, or we can do a trach. And I was thinking, if you're asking me, I think, I will do a trach and just bypass all of that. But the thing is, no one ever said tracheomalacia until we actually decided to have the surgery. The anesthesiologist was like, it's, he has tracheomalacia, the collapsing of the trachea. And so I go looking, I'm sitting in their offices, so my cousin and I were like, well, they walked out, we looked it up and everything, and sure enough, it's a symptom. But I noticed that this is not a symptom that you're seeing.
That's interesting. That's an interesting point. So we see a lot of hypotonia, so decreased tone of the muscles. Mm-hmm. So tracheomalacia is related to that. So it's sort of decreased muscle tone in the airways. Mm-hmm. So definitely something we see. We also see what's called central hypoventilation. So you can have decreased breathing, right? That's due to differences in the airway or the brainstem. And the brainstem is involved certainly in some of our disorders, including PMD and PMLD. So we do have patients who end up, needing trachs, or BiPAP is a little bit more common now. So to provide a little bit of that extra support, as well as the backup rate when their breathing slows down.
Right, exactly. And he didn't develop that till he's 16. But yeah, I was just curious about it.
Yeah, no, good question. Definitely see it. Mm-hmm.
Does anybody else have any questions? All right.
So in your PMD presentation, there was limited amount, mostly was less than 40 months or what have you. Mm-hmm. Was that simply because you didn't focus on older people, individuals? Okay.
This is a little bit biased. I think the early childhood group is a bigger proportion of patients that we... And that's a little complicated, right? Because we have biases. If everyone in medical school learns that PMD is a disorder that presents in the first 6 months of age, then, some of our older adults perhaps are not even being diagnosed because it's not on the adult neurologist's mind that this could be a disorder. So as we're learning more and more, I think we're recognizing older adult presentations increasingly, and there's a lot of work that we need to do there. Mm-hmm.
Awesome. Anybody else have any questions?
Thank you.
A round of applause for Julia. Awesome. Well, then we can go ahead and start moving on. We'll move back to the other slide deck. Perfect. And through the actual numbers of the data in here, but for the sake of time, we will go ahead and move on. I can share these afterwards. Do want to bring up our friend Grace Hobson to talk about a couple of the more genetic sides to PMD and PMLD. And then we'll get you set up.
On board? No? Yes? No? Yes. Okay. Here we go. Okay. So I'm going to talk about the genetics of PMD and PMLD today, and I wanted to keep them separate because it's a little bit different. So let's see. He told me how to do this. Okay. There we go. So PMD first. And so these are some of the PMD patients that I know and love. And when I was trying to collect pictures for slide presentations, I would always ask families to give me pictures of, events, things like the party above and enjoying things in the ocean. And this is actually one of my very favorite pictures of a PMD child because he's just having so much fun in the waves and doing things that other people do. So the gene that causes PMD, or the PLP1 gene, is located on the X chromosome. And oops, I'm going to do that every time. All right. So this is a cell with a cutaway, and the genes are located on chromosomes that are in the nucleus of the cell. And there are 23 pairs of chromosomes. And when scientists work on the chromosomes, they can do some staining that will band the chromosomes so that they can tell which one is which. And the largest pair of chromosomes is chromosome 1, and all the way down to the smallest, it's 22, pair 22, and then you have the X and the Y chromosome, and this is a chromosome set from a male patient, so you do see an X and a Y, and a female, of course, would have an X and an X there. So this is the banding pattern for the X chromosome, and the PLP gene is located at band 22 on the Q arm, or the long arm of the chromosome. And so we need to look at X-linked recessive inheritance for PMD. And this is a carrier mother who has the affected gene on one of her X chromosomes, and the affected gene is here in blue, and she has an unaffected gene on the other X chromosome in white. And when you make all of the combinations of the eggs in the sperm to create their children, you see that the female carrier mother passes her affected chromosome along to a portion of her children. And these you see in the solid arrows, and then the non-solid arrows are the rest of the chromosomes getting passed on. So a carrier mother and an unaffected father will have 1 in 4, or 25% of their children will be an unaffected son, 25% a carrier daughter, 25% an affected son, and 25% an unaffected daughter. And so how the genetic code works. Didn't mean to do that. We're back up. The pointer is just below the changing the slides, and so I have to kind of get used to this. I haven't used one of these before. Okay, so my pointer. So we're looking at just basically the outline of a cell here to show where things are. And so inside the nucleus, we've stretched out the chromosome so that you can now see the double-stranded DNA, and it's making here a double-stranded helix, and you've probably heard about the double helix. So DNA comes in 2 strands, and there's a top strand with the genetic code on it and a bottom strand with the genetic code on it. And this is where the bases are. The bases are where the genetic code actually resides. So bases can be A's or G's or C's or T's, and then the A's always pair with a T, the G's always pair with a C. And next, the RNA is made from the DNA as a template. And so here we see the RNA stretched out at a point where we can actually see the bases, and it's the bottom strand of the DNA that gets read, and the top strand that gets made. And so the A's and G's, A, G, C, is the first 3 bases of this particular strand of RNA. So the RNA gets made, and there are, it has an intron-exon structure some of the RNA doesn't code for protein. And you actually have to get that spliced out of the RNA. And I talk about this because there are some patients who have variants that are involved with the splicing of the RNA. But here you see that the green little pieces are not in this message RNA. And then this message RNA goes out to the cytoplasm, where there's translational machinery that attaches to it and reads the code along the messenger RNA. So every 3 bases of the RNA codes for an amino acid that's in the protein. And so this little machine runs along the RNA and it adds a protein or an amino acid at each step. And then the protein folds. And as Julia was talking about, it gets transported to the membrane, and that's where the PLP protein is active. So for actually how the coding works here, if you look at that C, did you see the A come in? That's a variant. Okay, we've got a C that changed to an A, and as the DNA replicates and makes more DNA, the bottom strand will also get a T because it has to base pair with what's above it. And then in the RNA, you see that the C gets changed to an A in the RNA, and the 3 bases right here code instead from the amino acid that was there, it now codes a threonine. So that's how the code system works. And Julia talked about some of the types of variants in PMD, and now I'm going to show you a little bit about the genetics and what that looks like. So she mentioned here we go again. She mentioned the duplications. This is a duplication. And so you see now we have 2 copies of the PLP gene. And whenever geneticists are talking about DNA, they draw it out on a line or, like this. But you have to understand what size you're looking at. How far down are we looking at the DNA? What level? How many bases, in other words, does this line represent? And when we're talking about duplications, so here's 1 copy and another copy with a PLP gene here and a PLP gene here, the size range that we're talking about is 100,000 bases to 5 million bases. We're not talking about just 1 base of DNA. And so these duplications include many more than 1 gene. So, there are lots of other genes that are included in there. And as Julia talked about, the mechanism for the disease is overexpression. So these cells fill up with PLP protein, gums up the works, and the gene can't do, or the protein can't do what it's supposed to do. And in the duplication patients, you have a moderate, usually, but also could be a little bit more on the severe end of the scale. Another type of variant, and this occurs, and I think I forgot to mention that the duplications occur in about 70% of the variants, although I was talking with someone earlier at this meeting who said that he thinks that this is actually much lower than this, and that what happened was in the beginning when we were first diagnosing these patients on the molecular level, a lot of the labs only did duplication testing, and so they picked up a lot more duplication patients, and so this may be actually a little bit lower number, down to like maybe only 50 to 60% of the patients instead of 70%. But here we have the so-called point mutations, and here we're talking about only one base of DNA that's affected, or maybe 2 or 3, and that historically has been, we've said, has been about 20%. That's what may be up higher, maybe closer to 50%. So on a report, a testing report, this is probably what the variance would look like. So the C for cDNA, and that is just the when you make a DNA out of the RNA, it's called a cDNA. So this is at the RNA level, and this is a variant at position 617, and it's a change of a T to an A. And then at the protein level, at methionine number 206, there's a change to a lysine. So this is just sort of generally, what it looks like. It's going to be a different base for every patient. And these are gain of function, as Julia was talking about. The proteins misfold, set up that misfolded protein response. All kinds of things happen, and these proteins cause the cell to be sick, or even to die, and that's what causes the disease. And it's the range here is from very severe to mild PMD. And so now for some of the less common variants and their disease mechanisms, we have the splicing variants. Remember I told you about the introns being spliced out and the exons being left. And here is a DNA drawn as a line with some things on it, which is what we like to do. And in this case, the PLP gene is about 22,000 bases, and this is just a portion of that. So I'm talking here about, instead of like millions of bases, this is a diagram showing maybe a couple of thousand bases. And this is how we represent a splice, or this intron being taken out of the RNA. And so a variant that involves splicing would be a variant, usually they're in the intron, and this one is at the +1 position in this intron. And this 191 would be the last base of the exon before it. And what happens is that missplicing occurs. And this question mark here is here because just from looking at the sequence of the DNA, you don't know what happens with this kind of variant. You have to go in and do the experiments, test the cells in culture, or sometimes we use blood to do the testing to find out what actually happens in this case. But it can cause a very severe to mild phenotype in the patients. And another type of variant that's only in about 2% of patients is a variant that there's actually no PLP protein, and Julia mentioned these too. And these can be caused by a deletion of the gene out of the DNA, and that's what I'm representing here. So this was a little segment of DNA that was up here in the in the patient's DNA, and then it's no longer there. It gets recombined out, when the cells are dividing. But here again, the DNA that we're representing here, this little piece is probably about 600,000 bases of DNA that gets deleted out. And so there's some other genes in there as well. There's another way that you can get no protein from a variant, and that is to have an early stop signal in the RNA. So at the end of your RNA, there's a stop signal that tells that little machinery that I was talking about that gets on and reads the DNA sequence. It comes down to a stop, and then the machinery falls off of the RNA. It doesn't go any further. And so if a variant causes a change and not to an different amino acid, if it causes a stop codon to be formed early on in the gene, then what happens is the machinery, the translational machinery, falls off of the RNA at that point early, and the cell has a mechanism to get rid of those RNAs, so that it's a cell integrity thing. It'll get rid of those RNAs, and the RNA is degraded. And when that RNA gets degraded, no protein can get made. So it's basically the same thing as having a deletion, no PLP protein made. And so there's a loss of function. These would be called loss of function variants, no functional protein. And these have most of them have sort of a mild phenotype, but it can, on occasions, be moderate. And then there's another type of variant that results in about 2% of the cases. And that is where, should be down here. That's where the variants are causing a form of PMD that's called HEMS, or hypomyelinating of early myelinating structures. And here again, we have our linear DNA, and this is from the PLP gene, like what I showed up here, which is probably a couple of thousand bases of DNA that we're looking at. And again, we're talking about splicing, and you get an alternative splice here because there are two donor signals that join with the same acceptor. This set makes PLP1, a form called PLP1. This set makes a form called DM20. Julia mentioned this as well, but it is caused by this alternative splice. And a lot of my work has been on this form. But what happens when you have these kinds of variants is that there's aberrant alternative splicing. And instead of getting mostly the PLP formed, you get mostly the DM20 form. And it causes a mild disease. And so the reason that I wanted to go through all of these different mechanisms of PMD is that we really have to think about what the mechanism is and what's happening inside cells before rational tests can be developed for PMD. And so I wanted to go through some of the treatment research that's going on, and Julia again mentioned some of these, but I'm talking now from a more genetic look at them. And one of the ones is the Orbit study. Courtney is going to be telling you more about that, but that one is actually the only one that we have that's in clinical trials, and it's for duplication patients only. And it's antisense oligos that bind to PLP and turn it down. So it's rational to use duplications for this. They actually designed it for duplications, because the duplications are one that cause overexpression. And so if you can turn down expression and make less protein, then that's a good treatment for this form of PMD. And I've heard some pushback from some of the patients in the PMD community about this, that, okay, you have a trial now for PMD, and my son can't be in it because he doesn't have a duplication. And so, that's making me unhappy. Well, all I can tell you is that, this is the most common form of PMD. And so what's a pharmaceutical company going to go after? The most common, form, where there are going to be more patients that they can have in the study. And also, they've designed these antisense oligos to turn down PMD, turn down the PLP protein. So you're not going to use this antisense oligo on a patient who has a deletion. It's not going to work. And so, there are reasons why the company, went after this as a first treatment. Well, so my question is, would these antisense oligos work on a patient who has a point mutation? Maybe. And so the PMD Foundation has awarded a grant to Paul Tizar to do a preclinical study. And preclinical study means it's not in patients, it's in cell culture, it's in mice. And so he's actually testing ASO therapies to try to turn down the misfolded protein that occurs in the point mutation patients and some other patients, and see if that's going to help the phenotype in those patients. And so, why would you think that an antisense oligo that turns down the gene is going to help these patients? Well, if you think about the deletion patients that have no PLP, and those deletion patients have a mild phenotype, and some of the patients that have point mutations have really severe phenotypes. If you treat with that antisense oligo that turns it down, maybe you make something that's more like makes more like a patient that has a deletion, which is, less severe. And so Paul Tizar is testing several of the types of mutations that are in the PLP gene to see if they will help, if he can turn down the expression of the protein. There was another study that was being done by Drs. Goldman and Osorio to test oligodendrocyte progenitor cell transplantation. And these are a type of stem cell, a progenitor cell. And they've studied these extensively in mice, and they work in mice. And so the question was whether they work in humans. And they teamed with Sana Biotech to do this study. They had gotten to the point where they were about ready to do a clinical trial in humans, and the company decided, unfortunately, to cut their neurology program. And so Drs. Goldman and Osorio were looking for another company to pair with and try to get this work done. And I haven't heard from them whether they're going to be able to do that or not. Sad thing, but it happens quite frequently in the pharmaceutical world. there's this proof of concept study on deferriprone, the iron chelator that Julia was talking about. And Dr. Wolf has actually enrolled patients in the study, treated them, and found that it doesn't seem to have a whole lot of effect in the patients that she's tested. So that's not looking too promising at this point, but she's going to continue to test those patients. For this presentation, I was doing some searches on the web and to see if there was just anything more out there. And I found this foundation, the In Lorem Foundation. And it said on the website that they were starting to do a PLP1 splicing program to develop, and their company is to develop treatments for patients with an N of 1 variants. N of 1 meaning only one patient in the world that has that variant. Well, they've expanded that to fewer than 1 in 30 patients in the world, and they're actually testing one of their treatments in a tub for a patient that's another leukodystrophy. If they get a successful treatment for the patient, they promise that there's free treatment for life. I don't know. I mean, the patients who have point mutations in the PLP gene, most of those, there are fewer than 30 patients in the world. So you might want to look into this. There's the website. It's possible that you might just kind of look into something with them. I don't know. You just have to see. You'd have to be able to pair with a physician in a major center where they can do the clinical trial at the center. There's also a group at the Leiden University Medical Center, and they are starting some preclinical studies toward a treatment in HIMSS patients. And they asked me to send them some skin fibroblasts from patients that they would convert into oligodendrocyte cell and then try to test them. And what they're working on is the HIMSS, the alternative splicing, and they are using antisense oligos that will cause the alternate splice to splice back towards the PLP form. And there's a grad student who's working on it who sent me her data, and they're pretty early on in this, but this Annemieke Ertsmuth-Ruth has sort of pioneered these N of 1 treatments, and she's pioneered it for splicing variants and has been very successful and is working in the Netherlands. So there's some really interesting things in the pipeline, you know, for the future for PMD research. And so now I'm going to switch to talking about the PMLD1 patients. And this is a couple of PMLD1 patients that I know and love, and that's about all the PMLD patients that I know because I'm really very new into this. And there are fewer patients that have been identified. So the PMLD1 gene is on chromosome 1, and of course the chromosomes are in the nucleus, and here's your spread, and this is chromosome 1. It's the largest chromosome, and if you look at the banding pattern on chromosome 1, GJC2 gene is down here in band 42 on the Q arm, or the long arm of the chromosome. And the way that this gene is transferred is by autosomal recessive inheritance. And so both parents in this case are carriers, and you see their affected gene in blue on one chromosome of each of the parents. And the affected gene is passed from both the father and the mother onto an affected child. And the ratios here are 25% of their children will be unaffected, and 50% will be carriers of one or the other gene, and 25% will be affected. And it's regardless of whether they're male or female offspring. And this slide should look familiar too. The genetics is the same, you know, from one person to the next. It's the same as what you saw in the PMD. And again, the double helical DNA, you get a transcript that I'm sure they're splicing, but I haven't studied the intron exons of these patients. And you get an RNA that goes out into the cytoplasm for being translated. And again, it's the same coding system where if that C gets changed to an A, then there'll be a T on the other strand after the DNA gets replicated. And the RNA is going to have that A as the first base of this codon. And instead of a proline, you would then have a threonine at that position. And just like with the PMD patients that have one base messed up, this is going to cause the protein folding, and then the protein can't do what it's supposed to do. So this is what the reports look like for a PMLD1 patient. You have to accommodate 2 variants, one from each parent. And so variant 1 looks like just one variant from the PMD patient, and variant 2 it can be another variant. It would be written in brackets for one variant with a semicolon, and then brackets for the second variant. Sometimes it can be the same variant on each chromosome, and it will be written like this. You write them both. You don't just say it has both. And there are cases when you're doing genetic testing where one variant's found, but a second variant isn't found. And that can happen if maybe the variant is doing something from an intron and causing a problem, and the sequencing isn't done in that intron. But if that happens and they find one and don't find a second one, they'll report it as a question mark when they report the variant. And this is just the basics. It can get more complicated. But when you have an autosomal recessive disease like this, and both parents are affected, it's probably because of the parents are actually related, and this can have an impact on population genetics. So if there's a variant that gets out there in the population and is passed on as in the the carriers have it, it gets passed on in the population, and eventually you're going to have two carriers that will have a child and have an affected child. But it can grow in that population. And so you find some places where there's a lot of consanguinity, which is what we call it when the parents are related. You have a lot of consanguinity in those countries like Iran and Pakistan and India. And we find centers of patients that have, all the same variant. And the prevalence of PMLD1 is going to be higher in those places than it would be in other countries. So this is the gene therapy that that Julia was talking about for the PMLD1 patients, and it's Dr. Cleopa, and now he's collaborating with Dr. Sena Estevez, and it's really kind of interesting. But just to the published paper that they were talking about was injection into the brain. And what they're trying to do now is just inject into the bloodstream and get the therapy to the brain. The problem with that is that there's a blood-brain barrier, and that's caused by the capillaries that are in the vein, brain, can't talk today, the capillaries that are in the brain have this blood-brain barrier around them. They're surrounded by astrocytes, and they also have this inner lining of endothelial cells. And the endothelial cells, it's individual cells, but they have tight junctions that don't let things in or out. And so they don't let things out of the brain and into the brain, and it's a protective device for the brain. You want to protect your brain. You want these to be there. But when you want to do gene therapy and you want the therapy to get out of the brain, there are things that you have to do to do that. And so one of the things that they're doing is trying to be able to give an injection and have it go to the brain and get out into the brain. And so to do that, they use these viral vectors, they call them, that have a capsid around a virus, and they take the insides of the virus out, so it's not infective anymore, but they put the DNA in there, and the capsid carries the DNA out of the capillary and into the brain. And Dr. Cleopa had been working on some capsids that would do that, but unfortunately, they would only do it for children that were 30 months or younger of age, and so the gene treatment would have to be done at a really very early age. But Dr. Sena Estevez has been pioneering capsid vectors that will actually get out of the blood, across the blood-brain barrier of much older people, older children, and so they're combining efforts and trying to convince. They'll try to convince the FDA to approve a clinical trial to do the gene therapy in older individuals, and to have this higher payload that Dr. Estevez Sena Estevez is working on. And so Dr. Cleopa wants to do a proof of concept preclinical trial in older mice, testing them to see if they can get that capsid to go across the blood-brain barrier and into the mice. And he thinks that that'll sort of tie it up with the FDA so that they get approval to do this test in humans. And he was looking for funding to do that, and Storm might have some words to say about the PMD maybe being involved in that. So this is a really very exciting and new thing that's coming up, and if it works for PMLD, it's going to work for other diseases. It's going to work for PMD, and it's going to work for other neurological diseases where you have to get a treatment into the brain. So it's really interesting. And so now I'm going to turn to genetic testing and the types of testing that can be done. First of all is carrier testing, and that would be for known variants and done for people who are at risk of being carriers. Prenatal testing can be done to determine if a fetus is affected. And one of the ways to do that is by chorionic villus sampling, and that's done in the first trimester, usually between 10 and 14 weeks of pregnancy, where a small sample of placental tissue is taken to prepare DNA for testing. And it can be done another way by amniocentesis, and that's later during the second trimester after 15 weeks of pregnancy, where a sample of amniotic fluid that contains fetal cells is used to prepare DNA for testing. And my advice to anyone who is going to be doing prenatal testing by either of these methods is to get with your geneticist well in advance and talk to the genetic counselor that will help you through this process and help to coordinate all of the things that have to happen to get the DNA sample taken, because this is not something you just go into a doctor's office and have them take a chorionic villus sample. It's done in a specialty setting, and then the sample has to get to the right place to have the DNA testing done. It has to be done all within the time that you need to know the test results. And so, like I say, get with your geneticist well in advance if you want to do one of these or the other. And then the other thing that you can do for family planning purposes is pre-implantation genetic testing. And that would be where the sperm and eggs are collected here in number 1. In number 2, they perform in vitro fertilization and set up the embryo cultures. And as the embryos begin to divide, doing cell division, they'll remove polar bodies or blastomeres from very early stages of the cell divisions and test for the variant or variants, variant for PMD, variants for PMLD, to see which embryos have the mutations, and then select the embryos in number 4, select the embryos that are negative for the variant, grow the embryos to the point that they can be implanted, and then implant into the female and get a pregnancy, hopefully, and then have child or children that don't have the variant. And so I believe Doug Harder is up next.
Yes, he is.
Okay.
Perfect. And before we do jump to that, does anybody have any questions for Grace? Yeah.
Yeah, it's either for Grace or the speaker beforehand. Apologize, I forgot your name. So much information. Julia, thank you. In some of the natural history studies or the other studies that have been done, have they ever actually looked at the mothers and any clinical systems that have been presented to the mothers? Because mothers go to the doctor, maybe they're having some symptoms that are similar but not as severe, and the doctor brushes them off, and et cetera.
I had a slide in here to show how sometimes the mothers are affected, and it has to do with a process called exon activation, and when in really early embryos, there's no exon activation, but as development goes on, this process occurs to kind of even things up, because males only have one X chromosome. So if you had two X's that were active, it would not be a good thing. And so one X gets inactivated. And in half the cells, it's the X that carries the variant. And in the other half the cells, it's the X that doesn't carry the variant. And so you get this, half and half situation. Well, if a PMD variant is a, one of the regular variants, a typical variant, the half and half situation kind of changes, because the X, as I said, there were half and half, but one X gets inactivated, and the X that is the one, I always get this messed up, the X that is the one that has the variant is a severe variant, and those cells are going to die, the ones that has that one activated. And so the brain gets populated with the cells that express the normal X chromosome. But in the ones that have variants that are very mild, I have to get this right. It's the ones that are, it's the same process that occurs, but the cells don't die. And so you get, the brain gets populated with both kinds, and so half of them are going to express this abnormal variant, and you can have females that are affected. And so I know that females often feel kind of left out because they don't get talked about so much, but no, I don't mind. So.
No, I'm so glad you brought it up, because I knew I wasn't including that in my slides, and it hurt me a little bit, because it's something that we've acknowledged even in the GLIA meeting, that we need to do a better job at studying. So yes, we know that mothers can be affected, like Dr. Hobson was mentioning. Paradoxically, it seems like mothers and females generally are more affected in families with where boys have mild disease for that process that Dr. Hobson was talking about. So the cells are just not sick enough to die off or inactivate. And yes, we need to study mothers.
And I have to say, this is a personal thing for me because I'm a carrier of cystic fibrosis, and I have some of the symptoms, but talking to doctors about it, they just kind of, blow it off. So, but there are organizations out there that are looking at the carriers, and so you could probably do some searches and find them. I gave a presentation about the carriers at one point, so look for that.
mean to cut you off there. I had a question from the chat as well, but it looks like we've got one more beforehand, so we'll get to the chat momentarily.
How long has PMLD genetic testing been readily available?
Well, as soon as that gene came out, our lab was set up to start it, and some of the other labs did too. But I can't remember when that first paper was published. Was it 2004? So it wasn't long after that before the testing was available.
Awesome. And then the two questions from the chat probably will be holding a mirror up to the foundation, but do we know of any grants for pre-implantation genetic testing for PMD? Or more broadly, I add that last piece there, just any grants for any type of pre-implantation testing?
It's not research, so I don't believe that there would be a grant
Like
for
I
it.
said.
It's not research anymore. It hadn't been for a long time.
Gotcha. Maybe a mirror up to us, something that could be looked into. But then the other question here is, does the type of variant affect how frequently the mother passes the gene on? Followed up with a note, my mom is a carrier, a splice carrier, has 3 kids, and all 3 of us got the gene.
It's just the luck of the draw.
It's the coin flip.
You know, the percentages are there. Yeah, it's a toss of the coin.
Awesome. Well, thank you. And thank you for, especially for this, as we didn't have some of those notes available. So round of applause to our dear friend Grace Hobson for being able to pivot on the fly. So we greatly appreciate you. And then next up, do we have Doug here in the meeting? And we'll go back to that original presentation as well.
Hey, Storm, can you hear me?
Yep, we can hear you. Hello,
Perfect.
Doug. How you doing?
I'm doing well. Thanks for having me, Storm.
Yes, yeah, thanks for being here. You know, this was really just to be the kind of flip side to what Grace was discussing here, is to talk about the personal side of really how those decisions are made for kind of the next steps. You know, more than anything else, what kind of influenced those decisions, and what were some of your driving thought processes behind it?
Sure. So my name's Doug Harder, and I'm pleased to be with you today. Talk about family planning. Wish I could be in the room with you, but to reveal a little bit of the outcome of our journey, and seems appropriate for a conversation on family planning, sitting in the car at the parking lot of one of my daughter's softball tournaments. So I am the father of 4 amazing children with my wife, Katie. Our oldest, Jack, was born with PMD in 2006. Following his diagnosis, we became involved in the PMD Foundation, first through connecting with other families, through family conferences, golf outings, and eventually I took on the role of treasurer as a way to stay connected with the PMD world.
He's awake.
With that as a brief backdrop, here's our family story. Katie and I were living in New York City, having been married for about 2 years when we decided we were ready to grow our family and try to have a baby. Getting pregnant for Katie was relatively easy, even if it didn't feel that way in the moment. And the pregnancy was uneventful. At 36 weeks, Katie went into labor, and after a long day of labor, eventually Jack was born. From the second he was born, it was clear something was wrong as he was struggling to breathe. Jack was whisked away to the NICU. After 16 weeks, 3 surgeries, including the placement of a trach and a G-tube, we eventually made it home with our son. At this point, after many tests, we were still without a diagnosis, but we had our son home with us. Katie put her teaching career on hold in order to be both Jack's mom and primary caregiver so that we could live just the 3 of us in our New York City apartment without any nurses. After so many weeks in the hospital, being surrounded by others, we were ready to be alone and in charge of our son's care. A couple months later, we visited a new neurologist who suspected Jack had PMD. This was confirmed by both an MRI and a genetic test. Learning about PMD at this point was very difficult for me to process, given the challenging prognosis for the disease. Leaning on each other, we were able to focus on our little boy. One of the most important things the diagnosis provided us was a community of people to connect with so we didn't feel so alone in this journey. In fact, it was on the PMD website at the time that Katie, using her mother's intuition, knew that it was in fact Jack's diagnosis while we were still waiting the official confirmation upon seeing another little boy who had the same froggy legs. She was right. Part of the diagnosis process involved talking to geneticists who introduced us to the concept of pre-implantation genetic diagnosis that Dr. Hobson just talked about. This was a new concept to us as we weighed the next steps for our family. When Jack was about a year old, Katie and I began to more seriously think about our options. To us, the options were 1, not having any other children, 2, getting pregnant naturally, 3, IVF with either full PGD or gender selection, or 4, adoption. As we had started to think about having a family, Katie and I knew that we wanted to have a bigger family, often centering our thoughts around 3 to 4 children. This did not change after Jack was born, so we were focused on the other 3 options as we saw them. This was a decision that we took our time on and allowed our faith to help us come to an answer. The key thoughts we kept coming back to were 1, the value of the PMD diagnosis, which allowed us to have a decision to make, and 2, how grateful we were for the incredible doctors and researchers who had made this medical intervention possible. We had taken Jack to the doctor. We were looking to get him the best care that was possible to make his quality of life the best it could possibly be. Ultimately, we decided that we wanted to provide that same level of medical care to our unborn children as well. We ultimately decided not to take the 1 in 4 chance to have another medically fragile child, so we opted to go down the PGD route. For those not familiar, as I certainly wasn't, here's how the PGD process works. You start with the traditional IVF cycle, which meant I had to give Katie hormone shots for weeks to have her body stimulate egg production. Those eggs were then harvested and fertilized by the IVF clinic. At day 3, the viable embryos were sent to the PGD lab, in this case shipped from New York City to Chicago to be tested for PMD. At this point, we are not testing for gender so that the impacted embryos could have been affected males or females, carriers. This was important to us since if we wound up having daughters, we did not want them to be carriers for PMD and have to face the same decision when they decided to start a family. The unaffected cells were identified and shipped back to New York City. 2 healthy embryos were selected and implanted. 9 months later, 2 healthy baby girls were born. In reality, I'm glossing over a lot of the challenges. Happy to talk about those, any if you have questions that came along with the IVF process, including a blood clot in Katie's jugular vein as part of the complications from the egg stimulation process. It was actually in the hospital on her 30th birthday that while Katie was being treated, that we found out that the IVF had worked and that she was in fact pregnant with twins. One of the reasons we decided to grow our families was that we always knew that Jack would be a great big brother, despite the limitations that PMD had on his ability to communicate, and that he would welcome the added activity in our house. This was evident from the first time we brought him to the hospital to meet his day-old new sisters, and continued as they would crawl over and climb up on Jack sitting in his beanbag. Katie and I felt like our family still wasn't complete, so we talked about having another child. So we decided to freeze the remaining unaffected embryos to keep our options open in the future. Jack had his share of critical health challenges over the years, but I will always remember the night of January 10th, 2013, as the 5 of us sat laughing in our living room, having a great time as a family. That night, Jack died peacefully in his sleep. Shortly after Jack died, Katie and I knew that our family truly wasn't complete and that we wanted to have another child. So we went back to the fertility clinic to begin the process again. By this point, the doctor felt comfortable transferring 1 embryo. This led to another healthy baby girl. Our youngest child healed us all in a way. While she never met her brother, which is very hard for her sometimes, we all see him in her. Olivia has such an understanding of children with special needs and even started an inclusion program at her elementary school when she was in the 4th grade. Our twins are now 16 and about to enter their junior year in high school, and Olivia is 11, about to go into 6th grade. While the route we took to get here was a million miles away from what Katie and I had thought before Jack was born, I know I couldn't be more fortunate for the journey we have been on in this amazing family I have been blessed with. Thank you for letting me tell our story. I'm happy to take any questions.
Sorry. First off, while everybody dries their eyes in here, because thank you, that really was, I'm a little choked up myself. Does anybody have questions? I can give everyone a few seconds to take a deep breath beforehand.
Yeah, I have a question.
Sure.
So my husband and I actually are doing consultations right now for the same IVF and genetic testing. And so we are really interested in all of this. And I was wondering if any of the embryos that came back as not carriers or not affected, were any of them male or were they all female?
We never found out.
Okay.
I don't know. I forget if it was on the testing report, but we asked, I guess, not to find out, when we were moving forward with the implementation. We kind of didn't want to know or pick whether we were going to have girls or boys.
Okay.
But I assume that is something that they could test for.
They
If they could
can.
if they could test for PMD, I imagine it'd be far simpler to test for the gender.
So the having them send like the samples to be tested, did they have to send it to a specific lab or was it able to just be like something that they could do more easily? Like we are kind of unsure if they're gonna have to send it to like the place that I got tested to see if I was a carrier or if it's not that complicated.
So when we did it, and keep in mind this was probably 17 years ago, so I'm not sure if it has changed, but it was a center that tested just for PGD, like did the PGD testing. So they coordinated with our geneticist who sent the PMD results so that they could look to then match the genetic testing of PMD with the embryos that we had sent for the pregnancy.
Okay.
And keep in mind, I'm a little nervous answering all these questions in front of Dr. Hobson, who knows far more than I do.
Well, thank you.
Thank you.
Absolutely.
Thank you. All right. Does anybody else have any questions for Doug?
And I'll offer this up. I'm sure Storm can give you my contact information if anyone has questions that they want to ask me, in a different setting, or if they think of them later. Feel free to get my contact information from Storm, and happy to be a resource in any way.
Perfect. Thank you so much, Doug. We really do appreciate you, and a round of applause. Awesome. Now, all
right. Thank you.
Thank you. Now, for the sake of time, we are going to move quickly. I am calling on my friend Courtney. We will go ahead and we can skip past some of these slides, move to the one for Ionis. I do have them all. We have, went too far. All right. I made a fancy name slide for you so you can see it, but that's this one. The light one is the laser. Oh, wait. I left this here. It's the last presentation. No worries. Almost. And then you've got a mic over there. Perfect.
Can you guys hear me okay? Perfect. Well, hi, everyone. I'm Courtney Bisbee from Ionis Pharmaceuticals, and I'm so honored to be here with you all today. At Ionis, I'm part of our patient advocacy team, and in my role, it's all about being a bridge between families, caregivers, advocacy organizations, and our scientists. And the goal of this is to really make sure that the patient and caregiver voice is always heard and always valued across the drug development continuum. So today I want to spend some time highlighting how patient advocacy really helps drive those efforts of collaboration between patient groups and industry, and then I'll wrap up with a brief overview of our PMD program, which includes our natural history study called Rocket and our phase 1b clinical trial called Orbit. So before we begin, just a quick reminder, the information that I'll be sharing today includes material about an investigational medicine which has not been approved by the FDA or any other regulatory body. This presentation is also educational, so if you have any questions about your child's medical care, please speak with your care team who know your family best. And the fancy lawyers made me say that, so now I can move on. So at Ionis, we're driven by science, but more importantly, and one of the things that drew me to this company, we're also driven by people. Our mission is to develop RNA-targeted medicines for serious and rare diseases, especially when there are few or no treatment options that currently exist. We've spent decades at Ionis refining our antisense oligo or ASO technology, and it's truly a privilege to be here and to learn from your community as we continue to evolve and shape our development program for PMD. So this slide shows just a quick snapshot of our neurology pipeline at Ionis. You can see we're actively researching potential treatments across a wide range of neurological diseases, from Parkinson's and MECP2 duplication syndrome to Angelman syndrome, and of course, PMD. So at Ionis, patient advocacy isn't just about engagement, it's about true collaboration and impact. At least that's our philosophy and how we approach it. Our team works really closely with advocacy organizations around the world to advance shared priorities that really improve the lives of those living with serious diseases. And a lot of these efforts, and in my work, focus on research, community education, health policy, or healthcare delivery, how people are accessing new drugs, and quality of care. And our goal as a function is to really make sure that the patient perspective, your perspective, is embedded across that drug development continuum to not only help inform the development of our programs, but to make sure that our studies are always community informed. To us, it's not good enough just to create a study. We need to make sure that that study meets the community's needs. So that's sort of our approach there. So this slide shows that philosophy visually. Our approach always starts with alignment. So we first want to identify shared priorities between Ionis and the patient community, and then work collaboratively to help advance those priorities. So as you can see here, not all priorities of patient advocacy groups will always align with Ionis's goals, but that's okay. My role is to really identify, okay, where does that overlap exist through conversations with leaders like Storm and Angie, and really focus our efforts on those areas. So for the PMD community, some of the key overlapping priorities that we've identified along with your community leaders include advancing research. So how can we support studies that address community need? Providing community education. How can we ensure that families have the right resources and information? And we also support initiatives that advocate for policies that improve access and care. So addressing barriers to diagnosis and treatment, and really understanding the diagnostic odyssey that you all are experiencing. And by learning these insights, we're able to look at our work and say, is there a way that we can make impact and help the community as an industry partner? So by continuously focusing on these priorities, patient advocacy in our function always aims to create meaningful impact together with you. So that's a little bit about what our function and what my role at Ionis looks like, and what you as community members can expect from our team as we continue to develop our PMD program in the future. And I hope it provided you with just a little bit of context as to how the two studies that I'll be discussing now, Rocket and Orbit, how those were truly community informed. So now I'll shift gears and give a quick overview of our two active studies at Ionis, our Rocket natural history study and our Orbit clinical trial. Let's see, is there animation here? Yes, there was. So Rocket is what we call a natural history study. And in this study, in our study, we're following the progression of PMD in boys with a PLP1 gene duplication to better understand how the disease is changing over time. And as Julia mentioned, in these types of studies, no investigational medicine is given. We're truly just looking at how the disease is progressing. So for our Rocket study, around 32 boys ranging in age from 6 months to 17 years are expected to participate. And families participating in Rocket, they come into the clinic about every 6 months for their visits, and those visits include general clinical assessments, MRI scans, and sample collection, like blood draws through lumbar punctures. And we really want to track biomarkers over the course of this 26-month study. Our Rocket study currently includes sites in France, the Netherlands, and Germany, but we're actively working right now to open up new sites in the coming year, additional sites in Europe, but also a few potential sites in the US. So we'll continue to work closely with the PMD Foundation and some of our other partners to keep you informed as new sites are activated for this natural history study if you're interested in learning more. And then we have our investigational trial called Orbit. So Orbit is a phase 1b open label trial, which means there's no placebo. So all participants will receive the investigational medicine. And the main goal of Orbit is to really evaluate safety and tolerability of ion 356. So in a clinical trial, especially in an early phase trial like this phase 1b, we're mainly trying to find out if a new drug is safe to use in humans and whether people can reasonably tolerate the side effects. So this is what we mean by safety and tolerability. So safety looks at whether the drug causes any harm, and then tolerability looks at how comfortable people feel when taking the drug, whether the side effects like nausea or headaches are too difficult to manage. So researchers use this information from this early stage trial to decide whether a drug, in this case ion 356, can move on to larger later stage studies like a phase 2 or a phase 3 trial. So Orbit is broken up into 2 parts. We have part 1 of the study where participants will receive doses of ion 356 during the MAD portion of the trial. And I don't like that term, it sounds very angry, but essentially a MAD study stands for multiple ascending dose. And this means that we're evaluating the safety and the tolerability of different doses in different cohorts of participants. Once the MAD portion of the study is completed, participants seamlessly transition into our LTE, or the long-term extension portion of the study, where participants continue to receive drug, in this case ion 356. So we had a really big milestone a few months ago. All of our sites for Orbit are now active, and this includes 3 sites in the US, 1 in France, 1 in Germany, 1 in Japan, and the last one in the Netherlands. So as far as next steps, our team, now that we have our sites active and running, will be monitoring participants through the duration of the study. And as soon as we have data, we'll obviously be sharing that with the community to let you know how things are going and what those next steps would look like. If you have any questions about either of these studies, please reach out to your child's doctor. You can chat with Storm to learn more, and you can also reach out to our team at padvocacy@ionis.com. And I think it's important to note here, we're so incredibly grateful to all of the families who have taken the time to participate in both of these studies to date. There's a lot of families who are taking the energy and their time away from work or loved ones to participate. And these studies are a huge commitment, and researchers, especially us at Ionis, we're not able to advance any of these programs without your guys' partnership, trust, and your child's participation. So I just wanted to say a heartfelt thank you on behalf of our program team. So that wraps up my presentation for today. Just to summarize, Ionis, we're continuing to activate new sites for our natural history study rocket in the coming months, and we're, fingers crossed, on track to begin enrolling those participants this year. We're also continuing to advance orbit with all 7 sites now active across the globe. And lastly, and I think most importantly, we're continuing to listen and to learn from all of you in the greater PMD community to really just understand your needs and your priorities and how we can show up for you as a really strong partner. What gives our work meaning is really knowing the faces and the stories behind the science. And that's why being here today means so much to our team. So Storm, thanks for inviting us to join you guys today. If you have any questions, I'm going to hang around for a little bit after the symposium, so feel free to chat or email us. That's it. Thank you, guys.
Thank you, Courtney. You don't have to move yet, but I'm going to move some stuff. Excellent. Ashley, can I steal you? Amazing. So, actually, we got 2 mics. Okay. Which means we don't have to play handoff for it.
Yay.
All right, all right. I don't need to introduce myself, but I'm going to because I'm on the slide. My name's Storm Greenway, the chairman of the PMD Foundation, and I'm here with my dear friend Ashley Dyke, who is...
Yes, I am Ashley Dyke, and I am the founder of the LCC Foundation. We are a foundation that supports another ultra-rare disorder called leukoencephalopathy with brain calcifications and cysts, and I have 2 sons that have been diagnosed with this disorder.
So Ashley and I work together on a number of projects with GLIA, who we've talked about quite a bit today. And because of that thought that, getting to see some of the other layers that go behind patient advocacy sometimes helps paint a fuller picture of all of the behind the scenes. And so I asked Ashley to be here because she is actually building the LCC Foundation from the ground up. She is the founder, she is the president, she is the one that has essentially just got this ball rolling. And so because of that, I wanted to talk through just the reality of advocacy in this capacity. So all kind of easy questions, but definitely here for more of just a discussion. So define advocacy. Like, what does the word mean?
Yeah, for me, advocacy is being the voice for my son, Ben, who has had his voice taken away due to LCC. And there are several others who have had their voice taken away from this disorder. And so advocacy for me is being that voice for them, standing up, standing in the gap between the doctors and our patients. It's now an identity for me. It's who I am. It's why I wake up every morning, and it's what I do. So,
and we've certainly been introduced to a lot of different kinds of advocacy. I think so commonly we hear the word and it is, what does it mean? Like we're fighting for something. But in the time that we've been in this space, and I'm sure that everybody in this room who has been around somebody affected, you've learned that advocacy can come through so many different channels. It can come through legal advocacy. As we had mentioned earlier, having to call representatives and make some noise occasionally. This can come from being an advocate in person and at that moment. It can be telling a doctor that they're wrong.
That's my favorite one. Right? Yeah.
And something that we will cover, because there is a right way and there is a less right way to do it. Because we still want to make sure that we're fostering the right relationships with said care team. But at the same time, there are going to be moments where pushing back is necessary. And it's something that, heck, I've got plenty of stories, but we'll move on for the moment. So, touched on it a little bit, but how did the journey begin from a not just diagnostic perspective, but also how did you start?
The foundation itself? Yeah. Okay. Because the journey's been really, really long.
Well, give some backstory to that as well.
Okay, backstory. Ben had a seizure when he was 5 months old, so this would have been in the fall of 2013, and had no idea he was a normal baby up until that point, no signs. It was just trauma, right? We wake up, I'm changing his diaper, it's a normal day, and he just goes limp, and I just remember calling my husband. I don't know what to do. This is, I'm so scared. Call 911. I'm coming home, like all the things. And several hospitals later, we don't have any answers. And I think, that was the moment that everything changed, and then it just went from there. But for a diagnosis, getting our diagnosis took 8 years. He was diagnosed in the spring of 2021. And then a year later, we were approached by our research doctor that there was a grant available to her and that she could apply for, but she needed the patient voice. She needed a nonprofit patient organization. And I, without hesitation, said I would do it. I would do anything I could if it meant continuing her research, if it meant getting the money she needed to continue doing the important work to save our kids. So that's why I formed it.
Done great work so far. I mean, it's the by any means necessary, and I think that is rarely do people operate in absolutes. But I feel like us as a broader group of patient advocates, we get to live that every single day. This gets to be the I would actually do anything, but what does anything mean? So right. I think just hearing that story alone absolutely was the by any means necessary.
Yeah.
Now, this one a little bit funnier, and I know one you and I had talked about, but like, what was that moment? The one where you had to be an advocate, you had to actually, rattle the cage because the answer that you got wasn't the right one. And I have a clear one in mind, so.
Did I share that one? I don't.
I have a clear one for
our
you. Situation.
Okay.
So I know everybody has one of those moments.
Yes, I had a moment very clearly. I was asking for an MRI for my son in St. Louis is where we're from. So St. Louis Children's Hospital, wonderful hospital. I'm just asking for a MRI from my neurologist, and he's telling me, but why? Because you have this diagnosis and there's nothing that you can do about it. I said, no, there is. Actually, if something's going wrong, or they see a cyst, which is a symptom of our condition, those cysts can be drained. They can be, a reservoir can be placed. And he's kind of arguing back with me like, well, that's not how I understand cysts on the brain to be, that they cannot be drained, or that, and I'm like, well, you need to talk to, like, I just got so mad. You need to talk to the research physician then, because she is asking that we get an MRI every 6 months, which, you know, prior to diagnosis, we were getting one every year. The longest we went without one was 3, and that's when we knew something was wrong, and that's where we could see the progression that had happened. But up until that point, it was little to no change, you know, and all that. And then, I had Dr. Frazier telling me, no, we need to scan every 6 months and keep an eye on things. If any symptoms appear, you know, so I'm just trying to relay this information to our local neurologist. And he was basically arguing back with me that that's not how he understood how it worked. And I'm like, well, this is what she's telling me. And so finally, I think I encouraged them to connect, and in the nicest way I could. And we got that MRI, so.
The connection is so unbelievably real. For us, our son River recently got a G-tube placement, and again, absolutely love our hospital in Kansas City. It's Children's Mercy. They're awesome. They have so many great programs. Absolutely adore them. A little bit of pushback against the GI team when it came to the messaging that we were getting from the Children's Hospital of Philadelphia, who really promotes the idea of don't wait until there's failure to thrive to intervene. Intervene to not have to deal with the failure to thrive. For feeding tubes, it is, you know, the messaging that we got initially was, well, you know, he's underweight, he's not on the chart, but it's also not drastic enough that it seems like it's making this massive impact. So let's go ahead and wait till we get to a little more like failure to thrive, and then give him the tube, and then make up the difference, and then get him on the chart. No. That was a moment where, and bless my wife Natasha over here, who was seriously, we pushed. I said, if you need to make a phone call to CHOP, we're happy to be here while you do. Right. But this is what we need. We really don't want to fly back out to Philadelphia to get this operation done. Can we go ahead and can we go ahead and get it? And to no surprise, River gets the feeding tube, puts on more weight in the last 7 weeks now than he has in the last year.
That's amazing. Yeah.
The last year. Yeah. It just there are times to push back. And it is still the GI team at Children's Mercy is thrilled at his weight gain. He's not on the chart, but he is making some headway. And even in that moment said, you know, they're glad that they did it. That's the important piece when it comes to the pushback. So that moment, I'm sure every one of us in the room has had that moment. It's okay. It is okay to push back. It is okay to know that we are, we're right in a lot of these situations. So, and you kind of answered this already, but like, how do you find care in this, not just with Dr. Frazier, but also in the broader sense for some of the more routine care? How do you find it? And more importantly, how do you address it to the new people on the care team?
Ooh, that's tricky. So I think, you know, with our disorder being so ultra rare, we do see a CP clinic, because that's the closest to, you know, what we can get at St. Louis Children's Hospital. And so we'll do wheelchair clinics and PT, OT, you know, all the therapies and things like that. But for anything routine, like the MRIs, now that, you know, I fought so hard for that one, and now I just don't want to fight every single time we have to do that. So we just go, we travel to DC, unfortunately, for all of the LCC related testing. And so we just do it all. We do Botox, you know, he's sedated, he gets the MRI, we get phenol injection, you know, we do all the things, blood work for research. anything they need, we do while we're there. It's definitely burdensome and needs to get better, but we'll work on. That's something that we have to work on as a foundation. I'll hopefully add it to the list of things that need fixed. So.
And I know for us and for a lot of us in this room, we have found leukodystrophy centers. A lot of us go to CHOP.
Yeah.
And I know a lot of the community has been to CHOP, but the same thing. It's to paraphrase Dave Manley, the chairman before me, is would you really take a Lamborghini to a Jiffy Lube to get an oil change? Probably not.
No.
This is if you're going to get specialty care, it doesn't hurt to go to a specialty doctor.
Right. Yeah.
So
yeah.
Finding that healthy balance.
Right. Yeah.
That's the tricky part.
It's very tricky, yes.
So then this quote from a dear friend of ours, Miss Melody Kaiser, when she had said this, I mean, and I have to read it right because this is one that she was like adamant needed to be correct. It's Melody. Everything is like, it's perfect. But rare parents deserve an honorary Dr. RD. So this is a doctorate of rare disease degree. They're the true experts, and their lived experience surpasses any academic credentials. I loved it when she said it. She said it at GLIA a couple of months ago, but from that point on, really, it kind of qualified the things that we go through as parents, whether or not we have the medical background, whether or not we have the clinical time. We have the all the time.
Yes.
And we're about to touch on this a little more with the caregiver panel coming up, so I don't want to take away from them, but from an advocacy standpoint, I find myself at least going into every doctor's visit with new care. I can't always make every appointment. Somehow she does, and it's remarkable to me. But I always try to go there when we're ever establishing new care, because have to come in and say, these are going to be the notions that you have. This is where you should probably get rid of some of those biases because this case is going to be a little bit different, and just setting that groundwork. So I know she said the quote to both of us as we were sitting together, so would love to hear your thoughts on this particular one.
Yes, it's truly validated all the work that we do, because I don't know how many times I would wake up and I'd be working on something and thinking, my goodness, do I need to go back to school? Do I need to go learn science? Do I need to learn how to run a business? Still think that every now and then, but it validated what we are trying to do, at least, like the effort that's put in along with caregiving, in running a household of 4 boys. So it was very validating. I truly loved that quote from her.
And it definitely lives forever with me. I know this is where we'll kind of end this out, so I know we're reclaiming a little bit of our time. But I think there's one other piece that we should touch on, and we'll probably touch on again in just a little bit, but the idea of trauma-based advocacy. There have been sessions on it. There have been multiple discussions on it. What brings a lot of us to this place is trying to make something good out of something that is really not good. For lack of a better way of putting it, we're all here because something forced us into this, or brought us into this, for people like our dear friend Grace, who just so happened to get assigned to PMD. But I know for a lot of us, and us as parents and us as advocates and those who are working in the nonprofit space, more often than not, we were brought here from a place that is not pleasant.
Correct. Yeah, absolutely. Definitely, right, the trauma of having Ben go through what he went through, and then watching him for 8 years and not knowing, finally getting an answer, and then realizing there's there's nothing. I think I had 3 days between diagnosis and then Dr. Frazier contacting me, and I was, right? Like, I couldn't sleep, crying all the time, off and on, all those 3 days. But when I knew someone was looking at his condition, I immediately was like, I have to help you. Like, this is my life's purpose now. Like, I just have to. And if no one else is doing it, and that's why I kind of held off a year. If she would have asked me day one, I would have formed the nonprofit. But yeah, it was a year. I kind of held off. There was another family I didn't want to upset because they kind of had the lead. But I did. I was like, I will help you. It's passion. It's what we do. Yeah.
I often joke that, as somebody who dropped out of college because I was changing my major too many times, was indecisive on what I really wanted to do with my life.
Yeah.
So to make the joke that, my purpose literally fell on my lap is what brought me into this space. And so, I think if there's anything that I can offer as advice, it's take breaks, try to disconnect when possible, because being in this space alone is difficult. Trying to actively move what feels like you're just trying to push back against a boulder. It's the picture of Sisyphus climbing up the hill, pushing the boulder, and it's like feels like the never-ending thing. So take the time.
I would. Taking breaks for sure, and trying to keep things as normal as possible. That's easier said than done, but yes. Yeah.
Well, then with that, Ashley, do you have any closing words for our group here in front of us today?
Oh, goodness. Thank you for welcoming me and letting me share a little bit about our story. And I hope that you keep doing what you're doing. I think it's amazing that you're here at this conference and working with the foundation and helping Storm. And it's just amazing. Keep doing what you're doing.
Well, thank you. Mm-hmm. And thanks for hanging out with us. Nice. And a round of applause for Ashley, everybody.
Thank you. Thank you.
If you'd like to, I'm giving you the chance. Otherwise, I am more than happy to. Okay. All right. Well, I wanted to give the opportunity because you have so awesomely put this together. So our next session is caregiver experience. So some personal insights, practical advice, and very much a diverse panel of different parent caregivers, circumstantially different, very much just in all of our experience, but we'll find that there are a lot of things that are shared. So we're going to introduce all of our panelists first, and then we will get going. First off.
Oh, there it is. All right. Hi, everyone. For those that don't know me, I'm Melanie Turen. I'm actually newly elected to the PMD board as the secretary, PMD Foundation board, so that's exciting. My son, Teo, is 8 years old. He has the connatal form of PMD. He's pretty severely affected. So in addition to the global developmental delays, he also has quite a few medical complexities, which you'll see as we all talk today. He lives with my husband and I in Chicago, so we're just about 20 minutes away from here. What Teo really loves is being outside. He loves swimming in the pool, specifically, and he loves Dr. Seuss books and Disney movies and just simple joys. And a fun fact, we thought we should all give some sort of fun fact, is that Teo was actually born in Germany. Christian, my husband, and I, we were living there at the time, and we moved here when Teo was almost 3 years old. And that photo in the bottom left of the slide is actually Teo with his entourage. My mom came, and so did 3 of her sisters to escort us over the ocean. We did a transatlantic cruise to get us from Germany to the U.S., which was quite the experience, but we survived. So if you wouldn't mind going to the next slide. Okay, well, for some reason, there's a photo missing. It doesn't matter. So our like I said, our journey started in Germany, and I wanted to go a little bit into some of the diagnosis and what happened with Teo. And interestingly enough, we already noticed when I was pregnant that Teo had symptoms. So I had too much amniotic fluid. I was ballooning up, and we were doing extra ultrasounds and testing. And they were noticing that Teo wasn't growing. He was basically staying the same weight and was severely underweight for his gestational age. And so it was decided that I should be induced. There was a specialist team there. He was, born through no complications during the delivery, but when he came out, he wasn't breathing, was immediately taken away from me, intubated, and sent to the regional hospital. I actually didn't get to see him until about 12 hours later because I myself had to go into emergency surgery. He stayed in that NICU for 14 weeks. He was intubated at first, and when that tube came out, that's when we all noticed the streetor. Like, everyone knew when Teo was awake because it was, you could hear him through the whole NICU. We started looking for answers. They thought there might be something with his vocal cords. So around 6 weeks, they put him under anesthesia to do an MRI and to do an EMG, which looks at his vocal cord function. And they told us he doesn't have vocal cord function, he has vocal cord paralysis. And they saw some abnormalities in the white matter during the MRI. And what was interesting was when he woke up from that, he had the nystagmus. And I didn't know what that was. It was my first child. So I actually sent some videos to my girlfriends and said, look at this funny movement that my son is doing. And they said, that's not normal. Like, you need to ask about that. So the doctors thought it's just from the anesthesia, it'll wear off in a couple days. And of course, as we know, it didn't wear off. So that started kind of our diagnosis journey. We were sent home at 14 weeks with a suspected diagnosis of mitochondrial disease. And they really said he probably won't make it a year. So enjoy whatever time you have with him. I'm sure a lot of people here have heard something similar. And but we think it's really important that you get a diagnosis. So they wanted us to have that for future planning. They had basically already dismissed Teo and said, you know, there's no chance for him, but for any other family members you might have, you need to know what this is. So we went to see another geneticist, and they gave us the suspicion of PMD, and we had to formally wait for the fish array. But of course, the first thing I did was Google, as we all did. And mother's intuition right away, like I knew that, just like Doug's wife, I knew that that's what he had, as in denial as I was. And we did receive the confirmation a couple of weeks later. And at that point, you know, when you read in the literature, it's like they're going to pass away in a year and 2 years, especially with his severity. So we just focused on doing what we could for him. We decided to get him his feeding tube. The picture that's missing is actually the first picture that we took of him after he got his feeding tube, because it was the first time in 7 months we saw his face with nothing on it. And it's one of my favorite pictures because he just looks like an angel. He's just so beautiful. And you can see in the photos here, you know, the struggles that he went through as a baby. You know, the top left is the very first time I met him, besides when he came out, and it was in an incubator and I could just put my finger in there. You know, the picture on the right is the first time I ever held him. That was the next day. Picture on the left, he had this ridiculous EEG, and in Germany they just like smash things on your face, and this poor little kid, he just looked so silly but also still so beautiful to me. And the middle picture is when we took him home for the first time at 14 weeks, and we were back in the hospital a week later. I mean, it was a revolving door for us for that first year. But he didn't die. He kept getting stronger. He kept thriving. I learned how to take care of him. I found a support group of other parents. I started doing research, and we saw him start thriving. And Storm, if you don't mind going to the next slide, these are things that he did in his life. You know, we were told he would never walk, he would never ride a bike. He did both of those things, you know, with support, but you see the joy on his face from being able to do those things independently. He loves being in the water and floating, and he's such an integral part of our family that, you know, we couldn't imagine it any other way. He has started declining in the past couple of years, so we've certainly noticed a progression of the condition. He has lost a lot of skills. He's had some new medical complexities come up. But despite all of that, every single day, he still smiles. And it's just such a testament to his strength and his courage that, you know, he doesn't know what he can't do, but he loves what he can do and what we do together. And I think I went over my time, but I thank you guys for that introduction and letting me share our story.
This is the last session. We go over, we go over. I think we've got the room for a little while afterwards, too.
Okay. You got one? Yeah. If it works.
No. Hello. Hi, I'm Natasha. My son's name is River. He is 3.5. So I went to let me start. My pregnancy, everything went normal. He was growing as normal. There was no worries with our doctor. Looking back now, there wasn't as much movement as there should have been. But regardless, at my 38-week appointment, I was told I was coming back that night and I was going to be induced. So I went home. I was on the floor crying. I was like, I'm going to have a C-section, which was my worst fear at that time. So I'm crying. I'm like, I can't do this. So we go to the hospital and I'm induced, and I was in labor for 36 hours. And I finally had River, and he's on my chest, and he's taken away in seconds. He's bright blue, not breathing. They just kind of take him away, and I can't leave the bed. I had, I can't think right now, epidural. So I couldn't leave. Storm was with me, and he was like, what do I do? I'm like, go with him. So I'm able to get in a wheelchair and go visit him, and we're pulled into a room by a doctor that gives us, bleak, 20% chance he's going to make it, very low chance he's going to make it. He wasn't taking the blood transfusions. Final hour, basically, it's taking, where we're able to get him on a helicopter, get him to a hospital that can give him the care he needs. And so I was released I refused medical care so I could follow him to the other hospital. And from there, he was under a cooling treatment, and that's 72 hours. And after that, Storm was finally able to hold him for the first time. And after the cooling treatment, he has an MRI. We're told everything's good. He's fine. He's going to be fine. And so we were celebrating. It's amazing. We get to take him home after 2.5 weeks. And we're following doctors because we were in the NICU. And our neurologist, I had mentioned to him the nystagmus, which I didn't even know what to call it at that time. He's like, he's not developing like he should. I think it's cerebral palsy, but I'm going to, we're going to do the test to find out for sure what it is. We're told it'll be a month, and we get a call back probably less than a week that the results are in, and we need to come in to hear the results. And he wanted us to wait till Friday. It was a Monday. We got in on Wednesday. And that's when we were told he has PMD. And our doctor didn't know much about it. He told us we would have 5 years, maybe, and enjoy the time you have. We learned quickly that's not the case. Not always. Here's some pictures of River. He is the happiest guy ever. He loves Miss Rachel, Miss Apple, Miss Monty, anyone that sings and plays guitar, he loves it. And his fun fact is his first ride was in a helicopter.
Before he ever went into a car, he was already flying.
Yes.
Hi, I'm Gina Jones. I'm a mom of 2 great young men, age 27 and 23, Garrett and Gavin. They're here cheering mommy on. We're from Chicago, and talking about my delivery or my pregnancy was totally normal. Reading everything, what to expect when you're expecting, eating right, walking, doing my thing, all totally ready for this thing. And normal vaginal delivery, and pushed him out in 30 minutes, and nice fat 8-pound, 3-ounce baby. And the only thing that he had that I noticed was that he had strabismus. But being a nurse, I knew, and labor and delivery nurse at that, I knew that that wasn't a big deal, so I wasn't worried. However, at 2 months, I noticed that his legs, every time I laid him down, his legs were shaking. And this is Garrett that I'm talking about, my oldest. His legs were shaking. I also noticed that that he was never interested in his feet. Kids play with their toes. He was never interested. He was muscular, breastfed, and just happy guy. But laying him down, like I said, and noticing the shaking, I decided to look up in my own books because I thought maybe cerebral palsy because of these muscular legs, but showing some weakness and stuff like that. But then I started thinking, somehow I don't think this is cerebral palsy. I don't know what it is, but I don't think it's cerebral palsy. But so I made an appointment with our PCP, told him what I thought and everything. And he said, well, Gina, I think you should see a neurologist. So, of course, at that time, and probably still now, when you're new at seeing a specialist, it takes forever. So at 4 months, we're about to see the neurologist, and that's when I noticed the nystagmus. And I was like, oh, no, this is not cerebral palsy. This is something neurological is going on, but this isn't cerebral palsy. So I sit with him. He assessed Garrett, and Garrett, he says, has cerebral palsy with something called spasmus nutans because of the nystagmus. And what color is his room? Is his room dark? Because he was born in December, he said a lot of times babies that are born in December aren't getting enough sunlight. So make his room brighter. Of course, he had a dark ceiling, and he did not have a light and all that. I'm like, the room is about going to sleep, you know, and everything. But let's see, as we're going through all this and getting his therapies and everything, we also noticed that he wasn't developing and getting better with his therapy. So they started thinking other things, you know, but without telling me. So I always tell the specialists, whatever you're sending to my primary caregiver, please, primary care physician, please send it to me. And in that letter, he's thinking Pelizaeus-Merzbacher disease. And I'm thinking, what the heck is that? So I'm calling my mother, who's also a nurse, and do you know anything about this disease? I look it up, and they say that the children usually pass away within 12 years of life. So of course I'm sitting in my bathroom just crying, like, what is this? I don't believe this. You know, and then why didn't he tell me? So of course I call him, he calls me back, and I'm mad, very mad, that you didn't say anything. You know that I'm a nurse, and I know how to be mommy nurse. If we've been partners, why didn't you say something? Well, I wasn't sure. So, you know, I forgave him and everything. And he turned out to be fantastic. He had polio and was very sympathetic to Garrett and Gavin, and working with him was great. When we finally got the diagnosis, I think they got tested for every single dystrophy. I commend Gavin. We were stuck. He and I were stuck like I don't know what. We gave so much blood. But Garrett was a trooper. So it was March of 2001. Find out for sure that he has Pelizaeus-Merzbacher disease. So that was, you know, it broke my heart. Then a month later, which I was, during this time, I wanted more children. We wanted more children, but we weren't really, we wanted to focus on this, what was going on with Garrett, before we do anything else, move further. But guess what? We had gone to Alaska. I think we got very comfortable. Next thing I know, in April, I found out I'm pregnant with Gavin. So what we did was get an amniocentesis. And first I was mad with my gynae because I'm like, we're gonna keep the baby no matter what. Right. And at this point I'm 35, so she's like, Gina, we gotta do a lot of testing anyway, genetic stuff and all that. But I want you to get this amnio so you can make an informed decision. So, you know, I got over myself, I appreciate that. Found out he was a boy, something told me he probably has PMD, I did. I was at work, she called me in, I'm like, you know, work, I'm not coming back. Because I just knew. So from there, I lived near the lake. I went to my spot on the lake and just sat. And it's like, oh, what am I going to do? What am I going to do? But, you know, I pulled my bootstraps on and did a lot of research. At that time, there was no Google. There was Yahoo. I found the PMD Foundation on the Yahoo group and everything and connected with them. And, you know, they were very informative. Matter of fact, I called somebody, and guess what? She was just bearing her last child. And I was so embarrassed. I didn't know what to say. I was like, you know, how old was he? I'm very sorry. He was, I think, 30 and everything. And I was thinking, well, that's actually good news for me to have lived this long. So I'm grabbing at all the positives, you know. But when Garrett and Gavin were diagnosed, our neurologist said 85% of their cells, both of them, are affected. Don't think that Garrett is going to be anything like Gavin. And that was for true. Because Gavin, he was very high functioning, you know, very happy, moving around, crawling. Never got a chance to really walk, but he did do some creeping. Gavin, however, was very low tone, and we were talking about that. And he just, you know, he had very bad eczema and everything, but he was still really happy anyway. Then the SHIT hit the fan. He got sick, very sick, with a cold, and had severe sleep apnea is what we found out, and was back and forth to the doctor and all that, trying to figure out things for him. And remembering those times was kind of crazy because I'm dealing with two. I'm getting told that Garrett, he's not meeting his milestones, he's not gonna walk, he won't be able to read, he won't be able to do this and that. So I wiped it out of my mind because I have Gavin who's got to go through the same thing, but then suddenly he's G2, he's got to have a trach. I'd already mentioned about the options they gave me to get to the trach. And then I noticed that he was vomiting breast milk even. And that just broke my heart. And this was about 4 months old he was. And when you're trying to start introducing food, and he couldn't take any of it. So I'm telling his primary care doctor this. He said, Gina, you have to wait till the sphincter can prove itself whether it's going to develop or not before we can do a G-tube. I'm like, well, what's going to happen to his brain, his heart, his kidneys? He's not getting any nutrition in because I can only breastfeed him because he can't handle any other food. He is vomiting everything, severe gastric reflux. So we dealt with it. He got very skinny. Garrett was, developing and doing his thing. But we finally got the G-tube, and like you said, after getting that G-tube, gaining weight and more active and all that. But at 4 years old, in and out of the hospital with Gavin, Garrett was always by our side. They never, ever they never said that I couldn't have Garrett with me. And I never asked the question either. He's coming. My mother said, act like you belong, and I acted like, come on, Garrett. He's sitting there supporting his brother always. And knew everything and still supports his brother and cares about him. But Gavin was something else. He got through it, 4 years old. Our primary care doctor was like, Gavin is so strong. I didn't know he was going to live this long. I said, well, I'm glad you didn't tell me, because we would have had a major discussion about that. And he said, well, what do you all like to do? And I said, well, we like to travel. He said, well, now it's time to be a family. Travel, do the things you like to do. And I said, hey, no problem, we can do that. Going back a little bit, when we were coming to ULF conferences and everything, and there was only like 3 families with Dr. Hodis, who was our specialist, and he was talking about stem cell research. I was pregnant with Gavin and had Garrett. I think my parents were taking care of Garrett, and I pulled him aside. I said, well, the research is fabulous. This is great, but what do I do now for my children? What can I do? He said, well, Mrs. Jones, I've known kids that are just tripped, and I've seen kids that are bedridden, and they don't live very long. But what I want you to do is keep them active. Physical therapy, physical therapy, physical therapy. And I said, I can do that. I can definitely do that. And so that's why you all when you see my pictures, like Gina's just out there, this is what I was told. So good nutrition, keeping them active, engaging them in school. Garrett was always in general ed. His kindergarten teacher, when he was in special ed, said that he's too smart for this. He needs to be in general ed with a caregiver. And he always had slurred speech, so we were questioned every time whether he could actually do these things. Garrett always blew them out of the water, always, and he's always been comfortable in his skin and everything. Gavin right behind him, he doesn't talk at all. I always felt bad having to do the trach, because I'm like, oh, I think he was starting to talk, and I just took his voice away. But Gavin talks, he vocalizes past that trach. They do have both communication devices, but Gavin prefers to use the body. Left is yes, right is no, left in between is maybe, and he does this when he doesn't want to be around or disappears or doesn't want you to focus on him. And then we give him choices. But he likes to speak directly. Even the doctors talk to him. One doctor asked him, can you touch his nose? He said, no. And I said, well, you asked him, can he touch his nose? Why does he want, he's not going to, he knows he can't, and he might poke himself in the eye. Rephrase your question. Do you want him to touch his nose? He's probably would say no anyway. But we've seen 13 different doctors. I've made my own care team.
We should have had them up here, all them big smiles.
Right, exactly. 13 different doctors, and because we see our doctors every 3 months or whatever, still things are happening. Gavin's trach came out on Wednesday.
Oh, no.
On last week, second time this year, last week we had to do a bronchoscopy. Turns out his tracheomalacia is past the trach, so the trach's not helping after 20 years of having a trach. So we might get rid of it. That's what we're talking about now. But my point of bringing those things up, things come up all the time. His trach is falling out like I don't know what. And I have never done CPR, but I've done CPR for the first time on my own child. After he was 18 months, and his trach came out. The nurse, I guess, got nervous. I had been up all night because I was a night nurse at the time, and she kept saying that the trach's coming out, Gavin's trach. I said, well, you know what? You all know what. Well, she didn't say it was coming about that Gavin's not breathing or something. I said, well, you all know what to do. I'm going to bed. I'm so doggone tired. And she was yelling, Gavin needs oxygen. And I was like, I got Dad, and there's a nurse. This nurse is going to bed because I'm not nurse anymore. But that didn't work. They kept yelling, and I came downstairs. Gavin's lips were blue. He wasn't breathing at all. And all I could think, he's 18 months, and when he had his trach put in, I remember the ENT was saying that, Gina, there's an emergency, an 18-month-old just passed away, and there's no reason why she should have passed away. The trach became dislodged. So that flashback came back, and I'm doing my CPR, full-blown CPR, couldn't feel anything. And I just kept saying that there's no reason for you to pass away today. There's no reason for you to pass, no reason, And I'm telling him, call this and that and all this, and, but he came back, coughed, and crying. I'm like, keep crying, you're doing great. So things happen, and I'm glad that I have the knowledge, the support, and the wherewithal to do it. I always call myself a Boy Scout. I'm always ready. Never know that I'm ready, but I'm ready. But with that, I'm not afraid to take them boating. Gavin loves being in the water. Rock climbing. Who knew? Someone invited, a therapist said, Garrett, you got guns, man. You should be rock climbing. And Garrett's like, okay. So we're rock climbing. Then they invited me to rock climb. I was like, what? Okay, I've never done it. I'm doing it. I'm doing it too. So, and that inclusion turned it all around. I'm in part of that inclusion. So we do a lot of family things. When we go on the boat, we're going on as family, but Gavin's the crew, and Garrett is manning the boat. He's doing his thing, and they're getting the lessons. They've been doing it for 15 years. We do cycling, hiking, camping, whatever. And it's an adventure. It's a big adventure, but under it all is PMD. Healthcare is a big part of our lives, but you talked about normalizing your life. That's what my plan was, to normalize as much as possible. So I can say this, that I got what I wanted and always thought I would in terms of children, because my thought was, what kind of people do I want to raise? And these boys are definitely that. They are humble, they're very smart, they have the confidence, they always try. What they can do, they do their very best, they always help. Garrett, when we come in and we're coming late, Garrett will take off Gavin's braces. He'll take off his shoes, help get him ready for bed, and I didn't even ask him. And Garrett wants, I just love those two. They're fabulous, and they love being together and helping each other. This, I just want to say about the mugs and everything. It started out as Band Together with 2 G's for Garrett and Gavin. Garrett told me when he was at 10 that he wanted to start a business to help take care of Garrett, he and his brother. So I said, okay, what do you want to do? And he said, I want to sell rubber bands. There goes Band Together 1.0. Right, so we sold rubber bands. We even went to the Children's Museum doing our little thing, But so years later, because that wasn't sustainable, during the pandemic, after taking art classes and art therapy, Garrett started doing digital art. And my sister was like, Gina, why don't you put that on mugs? Those are great, and it's great color. I'm like, I know, right? And so I tried them on mugs. My family was the focus group. They fell in love with them, so we started selling them. We did a website and all that. And one of my biggest supporters is Judy, bought a whole bunch of them. I was like, oh, thank you so much. But, that's our community. We support each other. And, making art basically out of nothing, making good out of bad, and these adventures out of nowhere, where you think that you might not be able to do anything. You can. The abilities of disabilities. the possibilities of disabilities.
She's hard to go after. I will tell you, as far as Gina goes, she was my first PMD mom that I connected with and met with, and she's walked me through this journey. She doesn't realize what an anchor she was at the very beginning when I first found out that Clark, my son with PMD, he has the classic form, duplication, that she kind of was like saying all the things she just told you. So it's like a flashback to that, and I needed that at that time, as we've all had those experiences. So Clark is my son. I'm Jennifer Martin Cutler. He's 10 years old, so that was his birthday party. As far as like the pregnancy journey, everything was pretty normal outside of realizing later that I really didn't feel him kick. He's my one and only. I went through like 6 different doctors, so like each visit would be someone different because you didn't know who would deliver your child. So I'd be like, I don't really feel him kick, but they're like, well, is it normal to you? And I'm like, yeah, it was normal. So, he went full term. He did end up being an emergency C-section, but really it wasn't until he was 2 months old, he was born with a cleft lip, just the lip part. So I knew right away he was going to have to get lip repair. So that led us to have to do, he didn't pass his hearing. He had a cleft surgery. He was going to have it 3 months old, and then we saw the nystagmus at 2 months old. So at 2 months was when we were like, oh, epilepsy. So we went to a seizure clinic down at Lurie's because we're from the Chicagoland area as well. And that was when they were like, no, this is neurological, and then we ended up getting genetics testing approved. And so he gets surgery at 3 months for the cleft, so we were going through surgeries not unrelated to that. So it was really because of the cleft that we even were able to get to the genetics testing and get it all approved through insurance. And then at 11 months was when we found out that he had PMD. Some of the things, like if you want to just go to the pictures, because I'll mention some of the other things, is I have to help feed him because it's such a fine motor test that he'll poke himself a lot, like talking about touching their nose and that sort of thing. But he can eat food. He is not on a G-tube. He doesn't need that. He has pretty good core strength. Actually, he's very good at pulling my hair. He's 10 now. He could probably take me to the ground if he disagrees with me on something. But that's our little community. You can go to the next picture, which probably is a little more defining. Clark is, as a 10-year-old, still primarily using a walker. But he also has a secondary diagnosis of autism, and certainly with cognitive impairment involved in that. So he does 5 days a week of ABA therapy in addition to this. So he'll go to school. We take him out of school about an hour early to do 2.5 hours a day of ABA. So, which we got approved it was during COVID, because I was like, he cannot do virtual learning. And he is in what we call in public school in a self-contained classroom, but he uses the walker. He's been using the walker since early intervention. So he was eager to pull to stand, but he cannot stand independently to this day and needs the assistance of the walker to move. But we are utilizing, I'm starting to call them Wheelchair Wednesdays, because he gains more independence through the wheelchair. He can reverse and say, no, Mom, I want to go over there and do those things. He is primarily nonverbal. He has a communication device, but I would say from a motor standpoint, it's been difficult to maintain that more high-tech device. He would rather do picture cues or buttons sometimes, but he's the most speaking, non-speaking, as we were talking about the utterances our kids do. He's been in and out of the room because I'm so all the rest are doing so great. Good jobs, Mom. But he's more behavioral. So physically he's got, we've done a decade of a lot of movement, and that was one thing that Gina taught me right away, was keep him active. And so now we're kind of crossing do we do the wheelchair more so he can be more independent and show us what he wants, or do we do the walker more? And then, he's still definitely diapers and toileting. We were hoping we could get him trained, and that's not going to be the case. But the fact that he can be vertical and I can change him is something I'm very happy with, because he's now 69 pounds. So that's as he gets bigger and I have to transfer him, those are things he can start, continuing to build. We have not seen regression. He's just kind of always stayed at this kind of unstable walking pace where he always needs the walker assistance there. So those are kind of the big things. As far as his things he loves to do is he's a rocker. So they had to get specialized chairs for him, because he was they were having to weigh down the tables and the chairs because he was rocking everything. So now they have a fancy Riften type of chair that he rocks in. And then if he can be on a swing or in a pool, that's his happy place. We've done aqua therapy. We've done hippotherapy, because those are what we can access and at that time afford, depending on if it's covered or not. So, and he's just starting to be more fine-tuned with his gestures. I talked about he has a dark humor, because he'll do something like, he's just recently been able to like hold himself up, you know, because head strength was an issue. And he'll like get out of he crawls at home, so he doesn't use his devices. So he'll like sit up and he'll reach out and he's like, I want a hug. And that's the first time he's maintained his core to reach for a hug, was just this last year. He has now since learned to also, I'm going to use you and I apologize in advance. He's also learned to do this when he doesn't want me. So I was like, oh, okay. So, and then turns around and wants to go to sleep or just doesn't want me involved. So I'm like, oh, great. You're more like your fine motor's improving, but you've got a pretty big attitude there. So, and his dad will attest to that, who's here. So, right. It's about the joy that our boys bring us and the journey we've all been on with a little bit different nuances, because all of us have to be kind of like our own doctors, right? And talk to each other on because even when we were talking about doing this panel, we learned so much about shared experiences that we've had and how we want to continue to do that. So that's all I'm going to share for now, because Jen still has to go.
Is this on? Yeah. I'm Jen Pack. You saw my Zach in here. Zach is the true completion of our 6-pack. We have 6 children, and he is our baby forever and always. Zach's story is very similar to Melanie's with Teo. We were born super critical. I couldn't tell you if my pregnancy was normal because I was in the middle of chaos with 5 other kids. My last 2 are 10 months apart. So I was chasing a baby, having a baby. I remember making my OB promise me I would be out of the hospital in 24 hours because I had a 10-month-old that had never been away from mom, that was still breastfeeding and super dependent. And 4 months later, they talked about us coming home. 5 months later, we got to come home. So unlike these guys, my heart breaks for them because their only experience is PMD. And we have 5 others. So our mourning has been a little different. They mourn that they never got to see it. I'm grateful we saw it, but I know exactly what we're missing because I saw it, 5 other times. So Zach is his own creature. He is as complex as they can be and still the brightest star in the room. He jumps from death's door to stable on sometimes a daily basis, way more than we'd like. He is TP independent, which is a challenge. That means he has a central line and runs an IV for all of his nourishment. His GI tract does not work at all. In fact, his own secretions create neurostorming and seizures. So we call him our drama queen. He loves that and has quite a sense of humor and thinks it's funny. But everything about him is complicated. He is BiPAP. He is oxygen intermittently. He is shaky vest. And we have all the fun equipment from every company that wants to give us something and charge us $1 billion, but he's changed our whole entire family. I can tell you all the stories about the diagnosis and about all the hospital stays. We call him Advocate Famous because we are here in Northwest Indiana, and we primarily use Children's Advocate in Oak Lawn. And there is not a floor on that hospital, and I am not kidding you, that when I push that child through in his wheelchair, someone doesn't say, Zach Pack, because we've almost died so many times, everybody's been there, okay? So he ranges from the NICU to the PICU to surgery they clipped his carotid. So the adult team knows him from the repair. And I think that's God. He has reached so many people. He tests me every single day, but he has brought so many people into our lives. And my kids are better people. So, there's a couple pictures up there. I didn't add a ton. That's my husband and I, and that's most of our kids. The 2 big ones were off doing something else. But our kids range from 25 to 7. Zach is 7. And this was a super hard year because in our diagnosis, we've heard repeatedly that children with conatal PMD and at the severities that Zach has seen make it 7 to 10 years. They kept telling us if you can get past 2, you should brace yourself for the 7 to 10 gap. And honest to God, we were blessed to see 7, but it was an emotional birthday because now we're in that realm of, maybe they're right, maybe they're wrong. I don't know. But we're super blessed to have him. As you guys saw, he was quiet in here. He was only quiet because I'm sure he thought they were talking to him. And he loves the attention. But yeah, that's our story.
And then through a little collage of some of the other photos that didn't make it to a few of these slides. Trust me, we have plenty more. The rock climbing one was one of my favorites. But
our dog Duchess, and they had the standing Danny, so that was really cool to be able to roll with that.
I'd be afraid that our dogs would yank River, because our dogs are.
We adopted her at 8. I wanted a cooled out, okay dog that was okay with the wheel. So we adopted Duchess. She had red hair just like I did at the time, and a mole on her nose just like me. And I was telling people that we got a dog that looks like me that's in love with my husband. But we adopted. I was saying we adopted an older female dog, because I wanted a dog, testing out whether we could have a service dog, whether boys could handle a service dog, and I wanted one that was nurturing and cooled out and not gonna pee in the house and all that. And Duchess was perfect. She was a great dog, so she didn't pull him over ever. She was really. She actually, that park, she would only bark when Garrett and Gavin were in the park, because Gavin's over here, Garrett is over there and zooming around, and Duchess, she wouldn't let anybody in that little promenade park that's right in front of our house, actually. And I would be embarrassed, very embarrassed, because I didn't understand at first. Why are you barking? No one's talking to you. No one wants you. Nobody's attacking. But then I realized that she was being a mom. She's protecting her boys, and you all can't come near. So I appreciate that about her.
In the spirit of continuing the discussion, because we could tell all of the stories of our dogs and River together, because it is the cutest thing. They've become extra protectors, but they are not calm. They are old, but they are not calm. Just seeing that made me think of the how they pull us. They're small, but I mean, dislocating shoulders bad. Anyway, so want to get the discussion going here. So the first thing that we wanted to talk about, as I'm tripping over a cable here, is self-care and mental health. So just kind of open questions here. I know we've got a couple of mics, so do pass them around as anybody has something that they want to put in here. What are your go-to strategies for de-stressing? How do we recharge?
I do early morning walks. I found a small little private park with some trees. I do my yoga and my stretching in there, and that is me getting away from them, because I always say they sniff me. Every time I leave the house, they smell mommy and they wake up. I'm like, I'll be right back. Mommy's got to go. I'll be right back. That's because it's Garrett that's always awake every time I'm about to leave at 6 in the morning. Just mommy's got to get her walking. So that's me getting my little thing in before my day gets crazy, because it's all about them once I get started. I also recently, for myself, to socialize more, because I'm naturally an introvert. I didn't talk this much until I had those two. but because I'm naturally an introvert, I made myself join a group of women who have children with disabilities. So I have to be honest with you, that got a little overwhelming for me. And I kind of stopped going to the group, but I connected with other friends one-on-one. I do better with one-on-one discussion, having breakfast and stuff like that every now and then. But I actually got a chance to go to St. Thomas with a girlfriend. Did a girlfriend group. Well, it was just me and one other young lady visiting a friend of ours in St. Thomas, a family friend. And that was for 4 days, and was the best recharge, because we were both going crazy and needed to get away. And I told her 4 days is not enough. I think I need about a month before I miss them. That's terrible, but it's true.
I'll go mine while you share your mic. For me, I was thinking about this a lot. So I work full time, and so although I'm also his caregiver, I'm also divorced. So there's 2 separate homes that Clark gets to attend. So every other weekend he goes with Dad, and that is a break for me. It's always one I know in advance that I have 2 days off, for that. So, and then also my work is another break for me. So I know that sometimes that's not possible for us, but in order for him to have insurance, I still work and I love my job. So I'm a school psychologist in a public school setting. So I have summers off too. So there's like really set breaks that I have. And those are kind of, I can't do yoga, and I definitely can't just like sit in my own thoughts because that gets scary. I forget to breathe in yoga. Like those are just not the self-care things I do, but I can mentally shift, every day that I work into something else that I really love and enjoy doing. So I would say that's probably like my biggest go-to for self-care is just having that divide.
I'm not good at self-care.
Yeah, self-care is very hard in this realm. We've recently, my husband has kind of pushed me a little to try and find a hobby because I've never really had a hobby, and we've recently gotten to rock climbing. So that's been our new jam to kind of decompress and just kind of get away from the day-to-day, and it's been really helpful so far. It's a nice just step away from life, and we've brought River with us one time so far, and he's a very chatty guy, so not sure how often we'll be able to bring him, but when he gets older, we're hoping we can kind of follow in your footsteps and get him on the wall.
Talking about taking the ballet class just so we can do it. Mhm.
So I'm actually going to kind of tell a little story and incorporate all three of those points because for me, it's not just one aspect. And I'm going to get a little personal here, a little vulnerable. So, we're all told you need to take care of yourself. You need to prioritize yourself. That's the only way you can be there for your kids. But you're thinking like, and when and how? And, yeah, that's great. I'd love to do that, but that's not going to happen. And so that's who I was. I was pushing through. I was working part-time. My husband was working full-time. We were taking care of Teo. We had some home nursing, and then COVID hit. And that hit home health really hard. We lost our nurses. My husband and I were trying to take shifts of who would be with Teo while the other one was working. We were trying to make up hours on the weekend. We were just going, and we kept thinking, this has got to be temporary. We're going to get a nurse. We're going to find more help. We just got to make it through this. And one night, I was in bed with Teo. He had woken up, which was upsetting for me for other reasons. He's not a good sleeper. And I was laying there, and my body started doing things that I didn't know what it was. And I thought, oh my God, what's happening? Like, am I having a heart attack? Like, something is going on. Somehow I managed to stay completely still until he fell back asleep, walked calmly out of the room, closed the door, and collapsed on the floor. Like, burst into tears, did not know what was happening. Christian's going, what in the world just happened? I mean, he didn't know, and I realized I was having a panic attack and just sat on the sofa, and he held me, and I just said, I just need to let it out. Like, don't talk. I just need to let it out. And, I calmed down, and that's when I said to myself, this is my body's way of telling me I can't keep going like this. I have to prioritize myself because when I'm like this, I can't care for Teo. And so that was a mental shift for us. We decided together that I would stop working. The idea was to take a 1-year sabbatical and find a nurse, and I would start seeing a therapist, which is a whole thing in itself. And we did it. So we started doing that. I went through a couple of therapists that didn't work. And this is a tip for those of you that are having trouble finding therapists. I actually went to our respiratory team who has a complex care clinic, and the social worker there had said, let me know if you ever need anything. I reached out to her and said, you're in this field. Do you know of anybody that can handle medical trauma as a therapist? She made a recommendation, and I've been with that therapist for 2.5 years. So when I started therapy, it was really intensive of dealing with the medical trauma from Teo's early years. Like when Gavin had his blue lips, we had that a lot with Teo. He would vomit and choke. I would have to resuscitate him. I would have to suction him. You do it, but it takes a toll on you even if you don't know. And there was so many repressed feelings that I had to work through because I would get so triggered so easily with little stuff now that weren't really important, but my brain had difficulty distinguishing what's a crisis and what's just something that you deal with. It was all messed up. And so the therapist had helped me a lot with processing those and coping strategies. And the other thing we talked about was, medication. I started with, doing blood work, and it turns out, low iron, low vitamin D. Those are things that a lot of us have. That was causing mental issues. But, I also had chronic anxiety and ADHD, and I didn't want to take medication, but I knew that I had to. So started seeing a psychiatrist, and now I'm on all the meds. I'm very relaxed. I'm very present. I'm able to process things as they come without getting that heightened awareness and without, having my adrenaline going. It's been a very beneficial process for me, but I really had to get to a breaking point before I prioritized myself. And, when people say take care of yourself, try to find a way to do it, because it is so vital to us to be able to care for our kids that we take care of ourselves.
I do have it on good authority. The wine also helps.
Wine. I'm a frequent participant in the wine game.
Like I said, good authority. All right. So next topic here, and this one I know to shift the tone of the discussion and almost a record-breaking pace. Let's go from the self-care to the things that might unfortunately drain that battery a little bit, which is proactively planning for the future. I think in every one of your all's circumstances, this is a really vastly different topic because it is going to, have more complexities and more nuance. So all the questions on here, how are we tackling difficult decisions regarding child's care? What steps have been taken as, this comes with age, financial, legal, residential considerations, and then how have we navigated or even considered end-of-life care?
I'll start. I'm ready. Now I'm ready. So this is a tough one for me. We met multiple times before this took place to kind of talk about what we were going to talk about and how we were going to talk about it and what everybody's views were. And you would be surprised how much this differed just amongst our group, right? Some of us, the planner over here, will tell you how she has everything done, and I can't even process that in my brain. Since the day of Zach's diagnosis, I have still not figured out how I'm going to bury my child. Or we're all thinking it. No one's saying it. I don't know how how any of us will get through that, right? I don't know how I could plan for that ahead of time. I didn't plan to cry here. This wasn't supposed to happen. So for me, and I'm sure some of you can relate, I call it the box in my head, right? And when we talk about self-care, my self-care sometimes is that I just can't do it right now. It's just not the day. It's not the time. I have other kids. I have other things in life. I just make myself busier. And I know a lot of you can relate to that. We put more on our plate because we can't process what it is we have to deal with. So it's the box in my head. And that's where it all goes. It's difficult decisions. And after the session today, if anybody has any insight on this, find me, okay? Zach's spasticity is out of control to the point that we are contracted in the knees, and I don't want to lose his ankles and everything else. And I'm a proactive mom. We're 2 days of PT, 2 days of OT, 2 days of chiro, 2 days of massage therapy. It's just not working. So you're going to meet with me afterwards and tell me what you got? So I just wish baclofen worked for him, and we're just not finding that. So difficult decisions would be, we're at a point where we have to talk about a rhizotomy. And I see these posts online, right? It absolutely cripples me to think about what my son's going to go through to have that surgery, right? And I have to look at the greater end of that and say, they tell me his ankles are going to be loose, and that once we get through the pain that the surgery is going to induce, that he's going to have less pain long term. And I know I'm not the only parent in this room that's had this conversation or like conversations, right? And here I am crying. But This was one that's been hard for me to put in the box because a Broviac was easy. He's going to die right now on the table if we don't do something. You know, a G-tube was easy. This is, will the pain and the outcome outweigh the process, you know? And I watch all of you, and so many of you have asked, has anybody done it? I mean, I think I've had one person. So it's hard, and I put it in a box, and I say, one day I'm going to deal with this, one day. But today, we're going to schedule a surgery. We're going to do the things, and we're going to do the best that we can. But not today. And so I commend you because she's planned it all out, right? I'm like, I don't even know if I want to see any of you after this happens. Like, I'm not sure I'm going to even be able to come out of my room when this happens. So I'm just going to take that one day at a time. I didn't mean to make everybody emotional. This is a tough one. But I think it's important to know that we all process it differently. And it's okay to plan, but it's okay to just go into it and say, I'm just going to have to face it when I get there. And until then, that box is going to sit right in the back of my head, and I'm not opening it until someone makes me. That's all I have. Who's next?
I would like Gina to talk a little bit about some of the future planning for children, but I wanted to jump in real quick since Jen, put me on the spot. I'm a planner, as she said, and we have Teo's end of life taken care of. We already started that when he was very young because we didn't think he was going to make that first year. We had an advanced directive with our palliative team in Germany. We knew what we wanted and what we didn't want. And we talked about that in a very rational way and said, these would be the potential outcomes. And we were happy to do it in a time, happy is not the right word, but we were grateful to be able to do it in a time when he was stable and we were calm. And so we made decisions about resuscitation, intubation, those types of things. And that continued on. We kept it even though he got much better. And when he started declining a couple of years ago, we reinitiated palliative services, which for those of you that don't know does not mean end-of-life care. That's hospice. Palliative is about comfort care and being at home. One of the benefits of comfort care is they do want to talk about advanced directives, and they do want to know if you've planned about these things, and they are experts in guiding you through this. And I had a friend who had sent me a TikTok on terramation. And I don't know, what's terramation? Terramation, in short, is human composting. So instead of a cremation, instead of a burial, the body goes into a vessel with organisms that help to decompose it within about 60 days. And at the end, you get soil. And this funeral home we selected is in Washington State because it's not legal in most states. And at the end of the 60-day process, we will have soil that we will be able to use to plant a tree, to plant a garden. They have a beautiful protected green space where, extra soil can be placed. And so we went through that process. We met with the funeral home. We met with the funeral home locally that would come to collect his body. And the other thing that was important to us was advancing research. So we also reached out to the Maryland Brain Bank, who is in charge of leukodystrophy and, brain donation. And so we are all signed up with them. So when that happens, which we know is going to happen, we make one phone call. And then there is a phone tree that happens. And the funeral home calls the brain bank, and then they call the funeral home in Washington. And we don't have to have a coroner come to the house, and it can be a peaceful, quiet time that we can spend with him without having to make those decisions. And that was for us very powerful. And I know that there are other people that can't face that, and that's okay too. But for me, it created comfort to know that I know what's going to happen afterwards. And as difficult as those discussions are, I really have a lot of peace of mind around it. And so, I'm happy to discuss with anybody, but I do think that if you feel that would be important to you, it's something to talk about, and a palliative team is a great way to kind of give you the guidelines and the appropriate paperwork and what do you have to do in order to really put those things in place. So that's me talking about, end of life. I think I'm the only one at this point that's gotten that far. I wanted to share that. I know that you have some stuff about.
Yeah, but I guess tagging off of you, I'm also a hospice nurse, in-home hospice care and palliative care, so I'm on the same wave as you are in terms of planning for Garrett and Gavin in that way, but we're not, I have to say they're still thriving, but the thing is having this stuff in place is important because you do have that one call, but it doesn't mean that you're looking at, death in the door. Hospice is, 6 months, that's happened, but my mother, who was in hospice, she lasted for 8 months, and as a hospice nurse, I was able to discharge someone alive. I saved his life. He got a chance to go surpass Christmas, and I'm not sure what happened to him after that because he did have HIV, but I was very happy to discharge someone alive. So it doesn't mean that you just let someone go and pass away or anything like that. You still get care, you get taken care of. You still, if you do need surgery, you still have surgery, but it's the difference between life-saving measures or life continuing is comfort. It's keeping the person comfortable, pain, taking care of the pain and all that, but still living, not waiting to die. You really live until you die, But I did go into hospice nurse because it was challenging. I was afraid of death. It was before I had children. And it was very rewarding for me to do it, too. So I have a better understanding of death and all that, and life and all that, and doing it day to day and having our adventures. And we don't have to be crazy every day, but just living, and enjoying the light and nature and things like that, and, the animals, whatever. But also, let's see, where are we? Financial? What the plan is, aging in place is my plan with nurses. This is what I would love to have.
That's my plan. We're not aging. We're just not.
I'm not talking about me. No. But the idea is what we do have a trust, so put our home in trust too, and it's for Garrett and Gavin, and having someone managing. And ideally having nurses there with them to take care of them because they're going to need care for the rest of their lives. As much as they do, I still have to change diapers. I still got to get the urinal. Garrett's, he's doing his thing, and still got to get them dressed. Still got to comb hair. Still I got to brush their teeth. And they're grown. But that's the reality of life. But that's the plan. The other part is. Also could be the reality is residential, might be more real. So I'm looking into I've visited different residential places like Misericordia. I've visited Markland already. I visited our home. I don't think it's in place anymore, but it was a set of homes that a woman donated for foster care. But she wanted one home to be dedicated for families with special needs. So that was something I was thinking about too. But residential, it might have to be a residential. The thing with the residential, they have to be appropriate and be able to take care of Garrett and Gavin's needs, especially Gavin having the trach and not talking, and just knowing that not everybody who has complex care needs is bedridden. And these two, especially Gavin, is proof of that. And but he can easily be bedridden because he's not going to demand to get out of the bed necessarily, Yeah. But once he's in that wheelchair, it's freedom for him. So going to the power wheelchair after dealing with all my I want him to have a manual wheelchair when I realized that he couldn't be in the walker anymore, and that was earlier than 10 years old. But when I realized. Got over myself about the manual wheelchair, and we got to the power wheelchair, but what it gave him was choice and direction, and added to that confidence and all that this is what we can do and where we can go. And Gavin loves to off-road. I found out all these things he loves to do. And he's got the perfect wheelchair for it, camping and night camping, whatever. He wants to do it. But those residential places have to have that in place to engage them appropriately. So, that's what we're thinking about. But in the meantime, those things are in place. But that's all I can say is that that's what I have in place. But right now we're at home, and we're dealing with nurses. Pandemic really took it away. I am a nurse. Guess what? Illinois was nice to let mom nurses be the nurse, get paid as nurses. And after the pandemic, just this, I guess a couple of years ago, they decided to continuing it. So I didn't want to, although I think it's great, but I'm like, I like being mom, yes, and nurse, but it's the paperwork that's killing me. The responsibility of getting paid being the nurse. It's not just signing in and out. But the next thing is the legal. We do have power of attorney of Garrett and Gavin. We did not do guardianship. Garrett seemed to be more independent. I was hoping that he would be able to live in a supported space. But I'm not sure. I'm leaving it to him of what he wants to do really on that front and how independent he wants to be. But right now it's power of attorney, Garrett and Gavin on healthcare and finance. And I found out you can get POA, if appropriate, for education as well. And that's important to put in place, guardianship, because as adults going to the ER, they can kick you out, whether he talks or not. Ambulance too. This I'm learning as I'm talking with the young adults with disabilities. It's not easy for them. It's not easy for the parents and all that. So I'm leaning towards guardianship, especially for Gavin, and we're thinking on Garrett. But it's an ongoing. It's not cut. This thing cannot be cut and dry because I don't know. For them, it's slow progressive, very slow progressive disease. And then next thing I know, Gavin's doing something I never thought he would do, and Garrett's swimming, and I never do, full face mask, and he's actually swimming. I never thought he'd be swimming, but it's I never anything they want to do or want to try, I let them do it. And I think that's it. Residential considerations. Financial, legal. We do have a lawyer that we have to stay with.
One thing you got to remember about guardianship is it's state specific.
On Mike, because that is an important comment.
This is my husband, Gordon.
Hi, I'm Gordon Jones. But the one thing that you got to remember about guardianship is it's very state specific. So in Illinois, the reason we haven't done guardianship for either Garrett or Gavin is it actually takes away every bit of their rights. So in other words, if Garrett and Gavin want to go outdoors, I have to either go with them or give permission to somebody to take them. They can't do it on their own, okay? They can't go and vote on their own, all right? This is Illinois guardianship. So like I said, it's state specific. So you have to look at your state to find out what the rules are that govern guardianship. And then in terms of guardianship, you also have to go before the judge once a year with an expense ledger, okay, that tells the judge if they get any kind of money, whether it's SSDI, SSI, whatever but if it's trust fund oriented, you have to give a dollar and cents breakdown of how you use that money for that given year, all right? And that's in Illinois. Like I said, it's state specific, so check your state.
Thank you for that call out.
I will just start with tackling difficult decisions. So at first, like he was mentioning earlier in conversation, was the G-tube. We did not want to get a G-tube until we absolutely had to, until a very kind doctor broke it down to us. You want to do it while he's healthy before we're failing to thrive, and it's just going to be a better healing process to do it. And as soon as we got home, we made the call, and the healing process, even with just the G-tube, which I know how small it is compared to some of the other surgeries, getting through it was like, how are we going to get through this? Like, this is just, how am I going to get through what's to come? Like, there's going to be way worse surgeries and healing processes that, but we got through it, and we're going to get through all of them, and you do what you can, and you do everything you can for your kid. And then I was going to say, as far as end of life care, we haven't looked at too far into that for River. We've been fortunate with his health so far, but, you know, as all of us know, this disease, you know, things happen, and it's always in the back of our mind, you know, what we're going to do. And also for us, like, if something is to happen to us, we have a plan for who's going to take care of him, and we have friends that will also be checking in to make sure he is well taken care of and getting the care that he needs, and just, you know.
Elaborating on that just a little bit more? I know the answer to this, but there's a lot that goes into sort of the legal framework decision, and, you know, do we have people to trust? What does that look like?
Yes. So the person that we have planned out has been to appointments with me, is trained on everything so far with River. I have a binder full of all of any info you need on River. I have it all together, and she's been along for everything. So it's a person that we trust with our whole heart. And then our friends are very, very educated on him. They live in Jersey, and when we go to CHOP, we get with them every single year, and they are like kind of like godparents in a way to River. So they just love him and care for him and want to learn everything they can.
There's so much kind of critical framework in that decision making for, you know, who do I trust, and to what end are there, like, guardrails? Basically, are we looking for, you know, if we were to all pass away and they were still to be around, would we expect them to become caretaker, or just make sure that he's comfortable where he's at? Like, that type of decision making is not something that we had. I know I'm interjecting myself into their panel, but not something that we had previously considered until it was brought up to us, is like, if something is to happen to both of you, what then? And that was a question that we had never really considered. So thank you for including that. I don't want to interject anymore here. Cool.
No.
Yeah. We'll move to the next one.
Okay.
All right. So next one here, celebrating joy and fostering engagement. What are the milestones and celebrations that we have? You know, how do we adapt to our environment or find new environments that adapt to our child? How do we make sure that things are accessible? What are some local resources or creative solutions that have been discovered? And then, what are your kids' favorite activities?
So I'll start with stuff for Clark. Like, there's the day-to-day, the swinging, the swimming, the things that we do to keep him active. But I would say even like I do fundraising for ULF a lot, and like reconnected with some people as far as helping those types of things. And I talk about inclusion a lot in the special ed program I work with, and including kind of Clark's story in those ideas kind of brings a lot of joy to me on using his story and like what we go through into both my work setting and then like the fundraising components of that. Like, we do things that are fun. Like, that's why I kind of don't want to ever be on a board and do some of the things that you guys have all done and are done with or starting to do. I just rather like raise some money and get some research out there and do some of that kind of thing and have a good time doing it. So you'll see some of those if you ever join some of our group. But like, those are the things I would say. And then like, you know, he's just really feisty. He's a ginger. So he's like always yelling at us and telling us what to do. And he has, you know, it's about building a village around him. I talk to his dad about this all the time, and Grandma's here and she's been very involved if you are on Facebook. But it's his village that like brings joy too. So we'll do stupid things like Wheelchair Wednesday, you know, is now something we like to do, where we just take out the wheelchair and see what we do. Or like we'll make up, like today's his half birthday. So like we'll cut a cake in half and say like happy half birthday, you know, like, so finding like the simple joy. And it may not be like, it has to be something he likes to eat, which right now are just like cheese puffs and cotton candy. I am, as Gina's feeding her children vegetables and I can't even have him touch one. But like, yeah, like I'm shoving Nutella. I was just telling his dad how he ate a half a jar of Nutella the other day. And his fine motor skills were so strong. I was like, okay, fine, you just do it. And he could do the whole thing, and he did kind of like get it all over. But anyway, you know, like it's finding though, and they are moments and they are glorious. And I think they're just like things we have to share with each other and know like that's like the best part of what we get to do and the opportunity they bring to us every day. So.
Am I next? You can, yeah. Okay. So the day-to-day, River loves to swing. He loves bubbles. He loves when his dad plays him guitar and sings to him. Every time he has a game and there's a guitar that comes up on his game, and every time it comes up, Dada. So, big fan of that. It's very sweet. When we're out of town, we try and find an art exhibit to take him to. We are a big fan of that. He can just kind of look around, and he loves the experiences that we are able to share with him. He also loves his sweets. He is a big sweets fan, so we typically go to the bakery a couple times a week, and we grab something just for him to enjoy, and he loves it. He is a big fan.
So fine motor skills, they're sometimes, able to get stuff to his mouth. But if he really, really likes something, he'll grab your arm and move your arm to his face. Like, give me more. I need it right now.
And I think that's one of the advantages of the G-tube is now he can eat for pleasure because you're not always worrying about the calories and the appropriate nutrition. You're able to get him that with the tube, and then he can eat the sweets, and you enjoy that. So for me, I wanted to talk a little bit about the middle point about how to make things accessible. And this is actually something that we were lucky enough to experience in Germany. They're called children's hospices. And again, hospice in this case is a misnomer because it's not an end of life, although they do provide end of life care at these facilities. But it's a respite facility for families and children with disabilities. And it's a residential facility that you go to a couple of times a year. They have 24-hour nursing care for the children, and the parents have their own wing where they have a bedroom and their own space. And if any of you have ever, taken your kids on vacation, you know that sometimes it can just be, I'm caring for my kid in another city. And I'm not really getting to experience the sights because they might not understand it. And just all the stress that leads up to packing for these trips and what if something happens. And so this respite facility was a place where Teo could experience new things. They had water therapy. They had horseback riding. They did excursions with the kids. They had art therapy. They had music therapy. But also for the parents, they had activities. They had an art center that you could go and make sculptures or make paintings or whatever you wanted to do. They had all these sorts of accessible things that you could do. But they also would bring in therapists and doctors once a week, and you had an ability to talk to these doctors that have so much experience with delayed children, and they would give you tips that you could bring back to your home caregivers and say, we would really like to try this medication, or we want to try this. And they were experts on this because that's who they dealt with day in and day out. And we were lucky enough to go there twice a year for the 3 years that we were in Germany. And, that's where we discovered that Teo loves being in the water. And we just got so much exposure. And I think it's a bit of a shame that there aren't those centers here in the U.S. But that truly was fully inclusive, take care of not only the child, but the family.
So there are some. Indiana has like Rosie's Place.
But are they a little? So the ones I looked up, the parents don't stay. It's more of a respite facility for the kids. This is just kids.
But they have all those.
Right. So they have the activities for the kids. But what I thought was so special about the ones in they're very prevalent in Germany, in the UK, and in some other areas of Europe, is that the family is taken care of as well. I could never drop Teo off at a facility and say, here's my kid. Like, he doesn't speak, and he needs medications every 2 hours, and he needs suctioning, and, go for it. That would never happen. So being able to be there and train the nurses, I always spent the first 2 days with the nurses so that they knew what the appropriate care was. They knew when he would communicate what it was he was trying to say. We really felt taken care of as an entire family. And that was so cherished to us, because the one time we tried to go on vacation before that, we just said, this is not worth it anymore. But we needed those breaks. And so this was truly an opportunity for us to get the breaks, and it was not only for the kids, but for the families.
So this one's kind of important to me for a different reason. I think that if you're sitting there asking yourself, what is my happy? What do I do? It's okay if you don't do. When I sat with these ladies and had these conversations, they have these great stories like you're hearing. We do this, we do that. We don't do anything. And I mean that in the most loving, passionate way, okay? Zach's excitement for the rest of this week was coming here. And you're lucky he tolerated sitting in here at the time that he did, right? And I started out with, he needs to go to Disney. He didn't care about Disney, okay? He was miserable. I needed him to be at Disney, and it took me some time to realize I needed it. He didn't need it. I needed it. I needed my other kids to remember their first trip at Disney with Zach in that memory. But Zach was miserable. We found that with most things. I bet a lot of you struggle with birthdays. I do. He doesn't enjoy it. He can't stand all those people in his house, in his space. He only wants Mickey Mouse or Coldplay on the TV. He doesn't care what you want to sing to him or what you want to eat. He wants to do his thing. And I listen to everybody have these great stories, but our kids are so different. We heard that from the doctor today. They're so different. And it's okay if your happy looks different. Because in our house, you know what a birthday looks like? A whole bunch of helium balloons. That's all he gives a crap about. He holds the string. It makes him gag repeatedly because of the tactile feeling of it, right? So, but he's so, and his eyes are so big because he sees them while he's going, bleh, 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 and gag and holding them over. But you know what? That's his happiest moment, right? And those stupid balloons live forever. So everyone now knows this is our happy moment, and you can imagine all of the gaggy strings hanging in our house for weeks. But that's how it looks for us, okay? One other thing I want to add, we are learning. It's a process. I don't have to always do something for him because he's happy right where he's at. If he sees me, and if I'm screaming at the other kids, he is so happy. He is laughing. He is engaged. Let me start dropping the F-bomb, and he is the happiest kid in the room, all right? If my older boys are passing gas and farting or fighting, he is so happy. And we're not doing anything but living, and that's okay. I know a lot of you, these guys don't have other kids, and that was different for me. We have a lot of kids, right? We lost a lot of life when Zach came into the picture. A lot. One, because their mom just couldn't even focus. I didn't know if my baby was going to be breathing the next morning, right? I put on 125 pounds. I know there are people in this room that can go with me and say, nobody had my back like that pan of brownies while I held that baby and didn't know if he was going to make it to the end of the week, okay? I mean, it's real life, right? And you do what you have to do. You get through it. We are learning to live again. So they're all probably sick of hearing Cruz, all right? But I'm going to say it again. You have to find your families happy. So for us, there's 6 of them, right? And Zach could give 2 shits about vacation. He really couldn't. I mean, he just, yeah. I mean, I'm trying to be politically correct, but he doesn't care. He doesn't care about Mickey. That thing was dancing in his face, and he was sleeping. He didn't care. But my other kids care, right? And I thought it was impossible. And I will tell you, call me, write down my number, find me on Facebook. I might cry, get a little emotional, but I will help you have the vacation or the trips of a lifetime because it's out there. We just did our first Royal Caribbean cruise. And I got to watch my whole entire family have the best time of their life. You know why? Because even when Zach was miserable, we just rolled him right down the hallway, laid him in the bed. He could stretch out, watch his movie, and none of my other kids had to stop what they were doing. They could eat whatever they want. They could drink whatever they want. They could sleep however late they wanted, and everybody was happy. For us, I hope that fit into this category because I got real passionate, but for us, happy looks different, right? And we sat stagnant for so long because the disease literally crippled us. It didn't just cripple my son, it crippled our whole family. Do you know how many pounds of IV fluid I took on a 7-day vacation? 50 pounds of IV fluid, just IV fluid. That's not counting the TPN, because if it goes bad, you have to have nourishment, right? But we did it. You know how many bags we took? 27 bags for 7 people. But we did it. That bellhop was like, I hope she got some cash in her pocket because, right? But we did it. So I'm here to tell all of you, even if your happy looks different, you just have to find it. And it's not easy. My husband can't stand vacation. Ask him. Like, he is the pack. We have to drive 2, sometimes 3 vehicles. Sometimes grandma has to haul stuff to the airport, but we're going. And you guys can too, right? Don't miss out. It takes a minute, but you'll find it. I should have been in a what? Listen, you know what I am? I'm a retired firefighter paramedic. Is that funny? Yeah. Yeah. See? You are too? See, listen, we're all lucky I'm not saying bad words. I thought, yeah. He said I could say shit right there. He said.
It's cool with me.
With us, what I found with young adults, teenagers, or whatever, everybody wants to give toys. And our children can't necessarily play. Because I know, because this one says nothing, and this one says something.
Do it, girl. He said, you try to shut me up, I'll show him.
But like I said, toys were just the bane of my existence because I have to get on the floor and play. Yep. Mom, you got to play. I'm like, oh, okay. But as they're getting older, the toy thing is just out. That's, it's a gift. It's just another piece of junk to me. So I decided that it's more of the experience. So Garrett and Gavin's birthdays are in December, Christmas is December, and then we got New Year's Eve, right? So the experience is staycation, a really nice hotel downtown with all our crap. Because we got tube feedings and we don't have to have the IVs and the TPN, but we got the diapers and all that other stuff that, whole stacks of stuff, but we do it, like you said. Yep. Then it's a play for Garrett or a classical concert for Gavin. And one of our classical concerts that Gordon took Gavin to see Beethoven with the serious classical people. Nearly got him put out because Gavin, like I said, he vocalizes and he loves to vocalize. He loves his crescendos and he loves those crash bang ends and he's going to say something, and so an orchestra hall is, you can hear everything. So Gavin's off to the side, they're ending, he does his thing with Beethoven at the end, they're done, and the piano player, guess what? He jumps out of his seat. So when we scheduled the next ticket to go, fortunately, me and Garrett, I stayed home with Garrett that day.
Next time we're bringing your boys up here too.
But it was. But the thing is, I got mad though because we ordered some next tickets, right? I'm a patron too, right? And they tagged us, Mrs. Jones. The last time you were here, there was a bit of a, and oh yes. I said, okay, all right. But we did pay. We're going to get free tickets. But we went, and they were very nice. I have to say, everybody at Orchestra Hall is nice. And we had a discussion with Gavin that we can do that outside or, in a better place. And he was great. He did great. But also we do Handel's Messiah, the do-it-yourself Messiah. The first one wasn't. We were at Orchestra Hall. Again, Gavin singing, but he wasn't the only one singing, and it wasn't the usual patrons either. So, of course, I had to hear about that. Because everybody walked past like, oh, he really enjoyed it. I'm like, you heard him all the way up there? We're way back in the back. But like our mother always says, act like you belong. I just smiled and took it, and we will be back. But so we do it yourself Messiah for Gavin, so we don't get put out. And Garrett, we do the plays and things like that. And one play that we took him to, he loved it so much that he cried. He didn't want to leave. So I thought that the security guard was going to come and put us out. I'm like, Garrett, you're acting like I'm abusing you. And I'm like, I haven't touched you. And he was only 3. He was 3 years old. But he loved it so much that he was overwhelmed. So he didn't know what else to do with crying. So the security guard was like, well, you can see all of the actors if you come out the back way here in the alley. Because that's where he was kicking us out. But we did get to meet all the actors and stuff. And that was very nice, a nice thing for them to do for us. But it's more about the experiences for us and everything. And what we have here in the United States is Give Kids the World. Garrett, I ignored him about this because I didn't want to go to Disney World. So Make-A-Wish asked Garrett what he wanted to do. He said Disney World. Well, I asked him. He said Disney World. And I ignored him for 2 weeks. I was like, no, he didn't say Disney World. I don't even talk about Disney World. But he said it, he wanted to go, so he went. They were fantastic. They hired our nurse to come with us. We had a little villa, Give Kids a Whirl. Yep. They had things for the parents to do without the kids, things for all of us to do, plus we were at Disney World. So it was the whole experience. And I was so doggone overwhelmed, but that gave me the know-how and that how we can actually travel. That was our first time actually traveling. With Gavin was, I think, 2 at that time. But he didn't care. Gavin did not care. He didn't, but it wasn't for him either, it was for Garrett. Yeah. So when Make-A-Wish actually called me about Gavin, I said, well, he's too young. I want him to enjoy it, and I want to get to know Gavin. And he may talk. Who knows? And it better not be Disney World. But so as I got to know Gavin, he gets this new wheelchair. He sits in his power wheelchair for the first time. He wakes up. He puts his head up. I put his hand on the joystick, and he zoomed forward. He could make choices. And he would run into the trees. He would go into the grass. He would run into the this and that. So he was just all over the place, and that gave them freedom. So guess what? Mommy got the fabulous idea. Gavin loves to be outside. Let's go to Hawaii. Oh. Let's go to Hawaii, because the reason why I thought about Hawaii is that, like I said, he loved being outside, and he loves water and all that, and we all do. And it's still in the United States, and I knew that we could have some accessibility. They rented a big van for us. They gave us the TomTom GPS, because this is 2008 to 2009. So over New Year's, we stayed in Hawaii for 10 days. And it was fantastic. The boys didn't want to leave. These are my winter babies who love winter. They're my polar bears, but they did not want to leave Hawaii. And it was a fantastic trip. We were all over the place. We don't stay put. They're like, you all are just different from everybody else. Would just stay on the resort. I'm like, I love the resort, but we want to explore. So we went and.
So the next conference, we're all going to meet somewhere really warm with palm trees. Here, we took a vote. Continue to address.
Yeah. Yeah, but I like the day-to-day. I like to mix it up. Some days we're going to do nothing at all. We like to, on the weekends, listen to audiobooks and discuss them or not discuss them, whatever. And Gavin doesn't like that, but he loves his music, and he loves to sing with his music in Gavi fashion. And then we all laugh and clap with him. His Gavi dance is doing this. And then Garrett is doing salsa sometimes with his girlfriend's aunt one time. The girlfriend just sat there, but looking at him, shooting daggers at him, but he was doing his thing. But, I try to normalize life, I guess, but it's more of the Jones life, what we do, and none of our lives are the same, but we try to do the best for our children. And the big thing is doing what you're paying attention to your children and what they like to do, and basically what makes them shine and gives them, I keep saying the word confidence because I can't come up with another word, confidence, and makes them feel whole. But we love just being together. And I think there was a question too, what do you do when you're not, who are you without your children, was a question that was asked one time with this group I went to. And I think we kind of put the same thing up here. But the thing is, I've incorporated, I love nature, love being outdoors. Swimming is really my big thing. But my children love doing that too. So I haven't lost myself. We've grown together, but there are things that I like to do separately, of course. But it's, I try to keep it as simple as possible,
Enjoy every day.
Yeah.
Before we move to the next, we're going to introduce the group. I have one last question for each of you. Very quick, very easy. I know, going off script just a little bit, but if you had one piece of advice to give to a newly diagnosed family, just small, simple, something to remember, kind of words to live by.
Paying attention to your child first. We're mad about the disease. It's devastating and all that, but don't forget your child. The person that's right in front of you does not know anything. All they know is you. And you have to be there and supporting your child and loving your child. So it's child first. See your child first.
I'll go next since I have the mic. Can you hear me? Yeah. Okay. I think just off the top of my head, when you asked me that, don't underestimate what the kids can do, what they understand. So often we do that just because they can't speak to us. We think they don't understand. And it's very clear. Teo will laugh when I'm telling a joke. And I'm like, did you understand what I just said?
Yes.
You know, and the doctors told us he would never walk. He would never ride a bike. Well, he did all those things. You know, he needed assistance, but we gave him what he needed. And so, yeah, it's really hard to get that news. But it doesn't mean that they're going to do or not do what the doctors say. So you don't underestimate them and give them the opportunities to do those things and adapt their world so that they're able to do them, because they may not be able to do it on their own, but with your help, they can absolutely do those things.
Go ahead.
Just kind of piggybacking, because my first thought was, don't put your kid in a box. Like, a doctor might tell you XYZ, but you know, look at some of these kids that we have in our community. They've surpassed what they were told, and you know, they're inspirational. So just don't put them in a box. Let them tell you and show you what they're gonna do.
You're not alone, and ask for help when you need it. You have a community, you know, from the people closest to you to the bigger, broader group. Ask for help. You're not by yourself.
Yeah, mine would be find your tribe. And I don't mean your family, your sister, your mother, your brother. These people are sitting right in this room. There are people in this room and people outside of this room that kept me, being very vulnerable. When Zach was diagnosed, there were days that I laid in bed at night and I prayed to God to take him and me in our sleep, because I could not live this life, and there was nobody that could take care of him like I could. I love my husband, and he's a hell of a dad. I said another bad word. But he can't handle Zach. And I didn't think I could live like that. And there are people, Melanie's one of them. Our children are so much alike that we literally, when she was in Germany, spoke in text all day long. We had an open text. When she was awake, she'd answer, and I would be awake later because our kids didn't sleep. Find that person. Find that family that can relate. Be that family. Look for families that their children model yours, because it's not hard to remember where we were when we were diagnosed. Help them get where you are now. You need your family, but your family doesn't get it. Your doctors don't get it. Your friends don't get it. They hide. I love my friends. They're not the friends that they were before Zach. I'm not the mom I was or the friend I was. But the people in this room are the people that are going to pull you through it. And I'm very passionate about that. This sit down is the start of, I hope, huge links. Everybody in this room that's watching it on video, we need each other. We need each other to get through this, because it's not an easy journey.
And would you look at that, a perfect segue.
That's why I went at the end, because we know this is my passion.
I love it. I love it. I love it. Melanie, would you like to do the honors?
Do you want to do it? I'm super excited about this. Can you tell? We started a new Facebook group. If you're not in it, get in it. If you know families that aren't there, invite them. We want a new atmosphere. We want an open platform. We want to connect families. Old families. I was going to point to you, and I didn't mean old. I meant families that have been through it. I know, see? Hey. I meant families that have been through it for a long time. We want you to connect with Gina if your boys resemble things that she's been through, because she can help. If your children are going through that severe NICU time, I want to be that mom that reaches out to the mom when she's sitting in the NICU, in the PICU, not when she's feeling like she can't go on, right? I want you to connect with more people, because you're so bright and you're so beautiful and your personality shines. You're an amazing grandma. There's some of you that I don't know. I want us to be there for each other. And so this group is super exciting. We're just getting off the ground, but get in there and get active, and let's get to know each other. I would love to see us become the group that meets outside of conferences, that can have trips and hang out, not just to learn things, but to learn about each other, to support each other. Because if you've met a family and you can relate with their children, you know there's nothing like that. There's nothing like having someone over, I mean, we've done this, where it's not stressful when their kid pukes all over your house. you have salmon real food blends all over the floor because your kid does it too, right? No one else gets that. So spread the word. I know, I'm off the chain. They're going to take this away soon. But spread the word. Storm got us started, and we're excited. We're so excited. So let's get everybody in there and start making things happen. Oh, and we have cool shirts. That would be my stress reliever. I make shirts. I put more chaos on myself when my son makes me crazy. So we'll be sharing the link soon so everybody can get their gear.
Well, the
QR
link is
code
there.
on the
Oh,
screen.
but
There's the link.
There's a link for the Facebook group, and Jen's going to post in there about the shirts if you want to order them. On a more practical side, something else that we want to offer, or we plan on offering, is monthly webinars that are educational, informative. For example, I have somebody that knows all about Medicaid waivers for all the states. She runs kidswaivers.org. She has already agreed to come on and talk. And that's so important for a lot of our kids, getting those waivers and getting that extra support. You know, we want to talk, have a therapist come on, a physical therapist come on and say, what's the correct way to pick up your kid so that you don't hurt yourself and you don't hurt them? What exercises can you do as a caregiver to protect your body as your kids get older, as they get heavier? So we want not just to be there for the rest of the community, but we want to be putting content out that's informative and educational for our community so that, you know, we can all feel like we're in this together. Oh, you got it. Gordon got a question.
I just have one thought that you might think about for the next probably 5 years. That is, what do we get a tract of land that's 400 or 500 acres?
You want to make a commune.
Yeah, well, that's exactly what I'm talking about. And we need to put it in one of the states. Now, your Medicare waiver person can really think about what might be the better state. But right now, I'm thinking southeastern Pennsylvania, southeastern Pennsylvania, northern Maryland, okay, and West Virginia. And the reason is not because of what's there. It's where we can go. We can go to Johns Hopkins. We can go to Kennedy Krieger. We can go to CHOP. Okay? We can even go to Boston if we had to. But the fact of the matter is, that's what's going to need to take place for all the children. It's not going to be just one set. And I mean, you know, lay it out. If we put all the balance sheets together, we could actually buy some serious land and put some serious buildings on that land. So therapy wouldn't be a problem. Pools wouldn't be a problem, okay? None of that would be a problem. But we need about 20 or 30 people to do that, okay?
I'm going to appoint you as the chairman of that committee.
Yeah. Well, you got a problem because I think I can get it done.
Hey, I'm looking forward to it.
That's the problem.
You're going to give us an appointment, an update next year.
Well,
if the foundation sanctions it, Angie has to sign off because she runs anything for committees.
Okay, well, yeah, but that's. I was the cook. See? No, but seriously, that's where we have to go, because when you talk about who's going to care for, everybody has family members and they all run the other way. Okay, so we know that's not going to work. Okay, the people that, you know, Jen's talking about, yeah, the people that are right in the community. I mean, the guys that were in the firehouse with me, all right, they still, if Gina puts out a call, they're coming, okay? So I know that it works. The question is to get everybody to kind of agree. I know you're not going to get everything you want, okay? It's not going to be red roses. It's not going to be near the water, but it could be, okay? I mean, it is what it is. But we have to look at the higher goal. And the higher goal is to make sure that everybody's child lives their fullest life, however long that is. All right? And that's what we do.
Yeah. Definitely put some thought to that one. So, for the sake of time, I've said so many different times today, we're 50 minutes over. We're going to go ahead and wrap up here. I do want to give a special thank you to everybody. First off, our panel, a big round of applause for our caregivers over here. And an extra special thank you to Melanie for putting this together and bringing this idea to life. It really was actually one of the driving forces that brought all of us here as an organization. And a special thank you to everybody else. A special thank you to Julia, who's no longer here. We're still going to give her a round of applause because she's here in spirit. A special thank you to Grace Hobson. A special thank you to Ashley Dyke. A special thank you to Doug Harder. A special thank you to everybody, all of our partners, because without you guys, we're just us. But together we're advocates. We get to fight for something, and we get to fight for something together. So a special thank you to each and every one of you for being here. And thanks for letting us steal away from the rest of the ULF time. And another special thanks to Ionis. I don't know if I mentioned you in my big list, but thank you guys as well, because you have been remarkable partners since my beginnings here. And thank you all for being the advocates that you are, because it really is a special job, and it is sometimes thankless work, but by God, is it rewarding. So, yeah. Oh, and we have tables at the dinner, so we can continue this conversation on. Our AV people, I'm sure, would like to get to the next thing. Another special thank you to our AV team, and a round of applause for everybody in here.